Vedantu Karnataka. Hegidira ellaru chenagidira. So please let me know in the chats if I'm audible and visible to all of you. Bega heli chat box alli if I'm audible and visible. Okay? Yes. Am I audible and visible? Okay. So students, ಇವತ್ತು ಏನು ಮಾಡ್ತಾ ಇದ್ದೀವಿ ನಾವು NEET classes ಮಾಡ್ತಾ ಇದ್ದೀವಿ ಅಲ್ವಾ? So we are started NEET ಮತ್ತೆ KCT classes simultaneously. So ನಿಮಗೆ NEET ಗೆ ಪ್ರಿಪೇರ್ ಮಾಡಬೇಕಂದ್ರೆ ಅದಕ್ಕೆ ಸಪರೇಟ್ ಕ್ಲಾಸಸ್ 5 o'clock ಸ್ಟಾರ್ಟ್ ಆಗ್ತಾ ಇದೆ and KCT ಗೆ ಪ್ರಿಪೇರ್ ಮಾಡಬೇಕಂದ್ರೆ 7 o'clock ಸ್ಟಾರ್ಟ್ ಆಗ್ತಾ ಇದೆ. ನಾವು ಮ್ಯಾಮ್ ಏನು ಮ್ಯಾಮ್ ಇಷ್ಟು ಮೇಜರ್ ಡಿಫರೆನ್ಸ್ NEET ಮತ್ತೆ KCT ಅಲ್ಲೇ? NEET ಸ್ಟೂಡೆಂಟ್ಸ್ NEET ಅಲ್ಲಿ ನಾವು ಬಯಾಲಜಿ ಓದೋ ಟೈಮಲ್ಲಿ ಇಲ್ಲಾಂದ್ರೆ ಫಿಸಿಕ್ಸ್ ಓದಿಸೋ ಟೈಮಲ್ಲಿ ನಾವು ಸ್ವಲ್ಪ ಡೀಪ್ ಆಗಿ ಹೋಗ್ತೀವಿ, ಆಮೇಲೆ PYQ ಸಾಲ್ವ್ ಮಾಡ್ತೀವಿ. ಓಕೆ, NEET PYQ ಸಾಲ್ವ್ ಮಾಡ್ತೀವಿ. KCT ಕ್ಲಾಸಸ್ ಅಲ್ಲಿ ಒಂದು ಪ್ರಾಪರ್ NCERT ಬೇಸ್ಡ್ ದ ಕ್ಲಾಸಸ್ ಆಗೋಣವಿ, ಅದಾದ್ಮೇಲೆ ಏನ್ ಮಾಡ್ತೀವಿ ಅಲ್ಲಿಂದ KCT PYQ ಸಾಲ್ವ್ ಮಾಡ್ತೀವಿ. ಸೊ ಅದೇ ಮೇಜರ್ ಡಿಫರೆನ್ಸ್ ಇಲ್ಲಿರೋದು. ಓಕೆನಾ? ಎಸ್, ಆಮ್ ಐ ಆಡಿಬಲ್? ಬೇಗ ಈ ವಿಡಿಯೋ ನಿಮ್ಮ ಫ್ರೆಂಡ್ಸ್ ಜೊತೆ ಶೇರ್ ಮಾಡಿ. ವಿ ವಾಂಟ್ ಮೋರ್ ಕರ್ನಾಟಕ ಸ್ಟೂಡೆಂಟ್ಸ್ ಟು ಕ್ರ್ಯಾಕ್ NEET ದಿಸ್ ಇಯರ್. ಸೊ ಅದಕ್ಕೆ ಈ ತರ ಒಂದು ಫ್ರೀ ಕಂಟೆಂಟ್ ಮಾಡ್ತಾ ಇದೀವಿ. ಆ್ಯಂಡ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಟ್ರಸ್ಟ್ ಮೀ, ನಿಮಗೆ ಡೀಟೇಲ್ ಆಗಿ ಎಲ್ಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. ಸಮ್ಟೈಮ್ಸ್ ಸ್ವಲ್ಪ ಡಿಫಿಕಲ್ಟ್ ಇರುತ್ತೆ ಲೈಕ್ ಫಸ್ಟ್ ಟೈಮ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮಾಡೋದಕ್ಕೆ, ಬಟ್ ಡೋಂಟ್ ವರಿ, ಮಲ್ಟಿಪಲ್ ರಿವಿಷನ್ಸ್ ಮಾಡೋಣ ಈ ಚಾನಲಲ್ಲಿ. ಬಟ್ ಎಲ್ಲ ಕಂಟೆಂಟು ನಿಮಗೆ ಫ್ರೀಯಾಗಿ ಕೊಡ್ತೀವಿ. ಓಕೆ, ನೀವು 1 ರೂಪಿನೂ ಪೇ ಮಾಡೋಕ್ಕಿಲ್ಲ, ಜಸ್ಟ್ ನಮ್ಮ ಚಾನಲ್ ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ ನಮ್ಮ ಜೊತೆ ಈ ಕಮ್ಯುನಿಟಿಯಲ್ಲಿ ಇದ್ಬಿಡಿ, ದಟ್ಸ್ ಎನಫ್. Okay, just be in this community with us. We will make sure that you are going to score really good marks. Okay, awesome. Everything is perfect. Okay, yes, quickly smash the like button and then we will start the class. Okay, so students, molecular basis of inheritance illi 40 marks pakka anta hakidini. You'll be thinking, ma'am 40 marks ah? Yes students, ee chapter ge general weightage nodidre 9 to 10 questions ee chapter inda barutte. 9 to 10 questions andre eno nimage already gottu. NEET alli 9 to 10 questions andre 9 into 4 illa andre 10 into 4. So maximum marks ee chapter inda nimage 40 marks. Okay, 40 marks from this chapter is pakka. Got it? So ನೀವು ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಮೆಸೇಜ್ ಮಾಡಬೇಕು, ಇಲ್ಲಾಂದ್ರೆ ಐ ವಿಲ್ ನಾಟ್ ನೋ ಇಫ್ ಐ ಆಮ್ ಲ್ಯಾಗಿಂಗ್. ಏನಾದರೂ ಪ್ರಾಬ್ಲಮ್ ಇದ್ರೆ ಐ ವಿಲ್ ನಾಟ್ ಗೆಟ್ ಟು ನೋ. ಓಕೆ, ಬಿಕಾಸ್ ಆಲ್ರೆಡಿ ಒಂದು ಸ್ಮಾಲ್ ಸ್ಟುಡಿಯೋ ಪ್ರಾಬ್ಲಮ್ ಇತ್ತು. ಸೊ ಕಂಟಿನ್ಯೂಸ್ಲಿ ನಿಮಗೆಲ್ಲ ಅರ್ಥ ಆಗ್ತಾ ಇದ್ರೆ thumbs up haki. If you have anywhere doubt, raise your hands or put any emoji. If you are enjoying the class, put fire emoji. Okay, first PUC class start ಮಾಡ್ತೀವಿ ಚಿನ್ನಿ. Okay, yes. Now here, basic introduction. Let's understand about this chapter very clearly. Okay, what is this chapter talking about and everything. Now students, nanne already Basava sir on basic introduction ಕೊಟ್ಟಿದ್ದಾರೆ, that genetics ಬಗ್ಗೆ ಮಾತಾಡಿದ್ದಾರೆ ಅಲ್ವಾ. ಇವತ್ತು ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲೂ ನಾನು ಅದ್ರ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಇನ್ನೂ ಡೀಟೇಲ್ ಆಗಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. ನಾವು ವಾಟ್ ಇಸ್ ಇಟ್? ಓಕೆ, ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇಲ್ವಾ? ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇದೆ ಅಲ್ಲ? ಫಾರ್ ಅದರ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಒಂದು ನಿಮಿಷ ನಾನು ಫೋನಲ್ಲಿ ಚೆಕ್ ಮಾಡ್ತೀನಿ. ಓಕೆ, ಸೊ ಐ ಕ್ಯಾನ್ ಪೀಸ್ಫುಲಿ ಟೇಕ್ ದ ಕ್ಲಾಸ್. ಇಲ್ಲಾಂದ್ರೆ ಒನ್ ಸ್ಟೂಡೆಂಟ್ ವಿಲ್ ಟೆಲ್ ಮ್ಯಾಮ್ ವಾಯ್ಸ್ ಇಸ್ ನಾಟ್ ಹರ್ಡ್, ಒನ್ ಸ್ಟೂಡೆಂಟ್ ವಿಲ್ ಟೆಲ್ ವಾಯ್ಸ್ ಇಸ್ ಹರ್ಡ್. ತುಂಬ ಕನ್ಫ್ಯೂಷನ್ ಆಗುತ್ತೆ. ಓಕೆ, ಒಂದು ನಿಮಿಷ, ಸ್ವಲ್ಪ ಇನ್ನೂ ಡೀಟೇಲ್ ಆಗಿ ಎಕ್ಸ್ಪ್ಲೇನ್. ಎವ್ರಿಥಿಂಗ್ ಇಸ್ ಪರ್ಫೆಕ್ಟ್. ಐ ಆಮ್ ಸೀನ್. ಓಕೆ, I am seen beautifully, voice is so beautiful, everything is beautiful. Okay, so basic introduction and discovery. Let's take example students. When you go for any functions, okay, illa andre when you go for any naming ceremony of a baby, okay, one small baby, ನೀವು ಹೇಳ್ತೀರಾ ಅಲ್ಲಿ ಹೋಗ್ಬಿಟ್ಟು ಅಯ್ಯೋ ಸೇಮ್ ಅಪ್ಪ ಥರ ಇದ್ದಾನೆ, ಸೇಮ್ ಅಮ್ಮ ಥರ ಇದ್ದಾನೆ. How do you say that? It is because you are saying because they look resemble illandre they look alike. How many of you can tell me in the chat box that ma'am I look more like my dad, ma'am I look more like my mom? How many of you? I look more like my mom. I am telling for me. Okay, I look more like my mom. Everyone who has seen me has told, hey, you look exactly like your mom, you walk exactly like your dad, you have complexion just like your dad. Now how are people able to make these predictions? Because they know that there is something called as genetics. Okay, there is a cell student. So ಈ ನೋಟ್ಸ್ ಎಲ್ಲ ನಿಮಗೆ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಶೇರ್ ಮಾಡ್ತೀನಿ. ಸೊ ವೇದಾಂತು ಕರ್ನಾಟಕ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಪ್ಲೀಸ್ ಜಾಯಿನ್ ಮಾಡಿ. ಅಲ್ಲಿ ನಿಮಗೆ ನೋಟ್ಸು ಏನಾದರೂ ಅಪ್ಡೇಟ್ಸ್ ಇದ್ರೆ ಅದ್ರ ಬಗ್ಗೆ ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಲಾಸ್ಟ್ ಮಿನಿಟ್ ಎಕ್ಸಾಮ್ ಟಿಪ್ಸು ಅಲ್ಲಿ ರಿಲೀಸ್ ಮಾಡ್ತೀವಿ. ಓಕೆ, ಓಕೆ, FF ಗೇಮರ್ ಇಸ್ ಟೆಲಿಂಗ್ ಮ್ಯಾಮ್ ಐ ಲುಕ್ ಲೈಕ್ ಮೈ ಮಾಮ್. Okay, so yes, even I look like my mom. Okay, so students, there is a cell. Okay, there is a cell that has all the informations. Okay, there is a cell that has all the informations. Okay, this cell is going to hold all the informations. Okay, it is going to hold all the information. Now when I say all the informations, what do I mean by that? That means, let's say how you are looking, okay, how your characters are. some of the smallest of smallest like features, like your nail shape. If you notice your mom's and dad's nails, you will be able to understand why your nails are like this. That is, even those small things, students, nim parents inda bandirodu. So adike navu ಏನು ಹೇಳ್ತೀವಿ, ಅದಕ್ಕೆ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಅಂದ್ರೆ ನಮ್ಮ ಪೇರೆಂಟ್ಸ್ ಇಂದ ನಮಗೆ ಸರ್ಟನ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್, ಜೆನೆಟಿಕಲ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅದು ಇನ್ಹೆರಿಟ್ ಆಗುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಈ ಚಾಪ್ಟರ್ ನೇಮ್ ಮಾಲಿಕ್ಯುಲರ್ ಬೇಸಿಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್. ಸೊ ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ನೀವು ಮೇನ್ಲಿ ವಾಟ್ ಆರ್ ಯು ಗೋಯಿಂಗ್ ಟು ಟಾಕ್ ಅಬೌಟ್ DNA and RNA. Now ma'am, what is this DNA and RNA ಬಗ್ಗೆ ಹೇಳ್ತೀನಿ. See, a cell that holds all the information. No, one second. A cell that holds all informations. Now who is this cell? You will be thinking, no? So then the cell is none other than your nucleic acids. Okay, your nucleic acids. This cell that holds all the information that you have, okay, the cell that holds all the information that you have is called as a nucleic acid. Okay, good evening, good evening. Yes dear, yes, it is good. Okay, so nucleic acid. Now students, nucleic acid banbittu, there are two types of nucleic acid. What are they? One is called as the DNA and one is called the RNA. So one is called the DNA, one is called the RNA. Okay, now students, ನಿಮಗೆ ಆಲ್ರೆಡಿ ಗೊತ್ತು ಮ್ಯಾಮ್, ನನಗೆ ಫುಲ್ ಫಾರ್ಮ್ ಆಲ್ರೆಡಿ ಗೊತ್ತು ಮ್ಯಾಮ್, ಇಷ್ಟು ಈಸಿ ಆಗಿದೆ. ಬಟ್ ದೆನ್ ಆದರೆ ಕೂಡ ಬರಿಬೇಕಲ್ವಾ, ಬಿಕಾಸ್ ಈ ನೋಟ್ಸ್ ನಿಮಗೆ ಲಾಸ್ಟ್ ಮಿನಿಟಲ್ಲಿ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಇವಾಗ ಖಂಡಿತ ಮೇ ಬಿ ಹೆಲ್ಪ್ ಆಗಲ್ಲ, ಬಟ್ ನೀವು ರಿವಿಷನ್ ಟೈಮಲ್ಲಿ ಈ ನೋಟ್ಸ್ ವಿಲ್ ಬಿ ಲೈಕ್, ಓ ಮೈ ಗಾಡ್ ಮ್ಯಾಮ್, ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಫಾರ್ ದಿಸ್ ನೋಟ್ಸ್. ಸೊ ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಲ್ಲಿ ವಿ ಹ್ಯಾವ್ DNA ಫುಲ್ ಫಾರ್ಮ್ ಏನು? ಡಿಆಕ್ಸಿ. ಓಕೆ, ಡಿಆಕ್ಸಿ ರೈಬೋ nucleic acids, correct? Deoxyribonucleic acid. RNA in students, ribonucleic acid. Ribonucleic acid. ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಮೇಜರ್ ಡಿಫ್ರೆನ್ಸ್ ಏನು ಸ್ಟೂಡೆಂಟ್ಸ್ ಇಲ್ಲಿ? ಸೊ ಐ ಟೋಲ್ಡ್ ಆಲ್ ಆಫ್ ಯು, ನಿಮಗೆ ಅರ್ಥ ಆಗ್ತದೆ ಇದು ಏನು ಥಮ್ಸ್ ಅಪ್ ಹಾಕಬೇಕು ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ. ಓಕೆ, ಯು ಕೆನಾಟ್ ಸಿಟ್ ಐಡಿಯಲಿ ಇನ್ ಮೈ ಕ್ಲಾಸ್. ನೀವು ನಾನು ಎಷ್ಟು ಆಕ್ಟಿವ್ ಆಗಿರ್ತೀನಿ ಅಷ್ಟು ಆಕ್ಟಿವ್ ಆಗಿ ನೀವು ಇರಬೇಕು, ಇಲ್ಲಾಂದ್ರೆ ನಮ್ಗೆ ಒಂದು ಕನೆಕ್ಷನ್ ಬಿಲ್ಡ್ ಆಗಲ್ಲ ಕ್ಲಾಸಲ್ಲಿ. ಓಕೆ, ಸೊ ಆಸ್ ಐ ಆಮ್ ಟಾಕಿಂಗ್ ಯು ಶುಡ್ ಕೀಪ್ ಆನ್ಸರಿಂಗ್ ವಾಟ್ ಎವರ್ ಯು ನೋ. ಓಕೆ, ದಟ್ ಇಸ್ ಹೌ ವಿ ಕನೆಕ್ಟ್. ನೋ, otherwise online teachers are going to be so difficult to connect. So ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇಲ್ಲಿ ನೋಡಿ there is something called as deoxy. ಇಲ್ಲಿ ನೋಡಿ ribo. That means there is something that is lacking here, that is present here. So deoxy means, from looking at the word only I understand that ma'am, it might be that they have absence of a oxy group, or let's say an oxygen group. Absence of an oxygen group, correct? Okay, absence of an oxygen group. ಇಲ್ಲಿ ನೋಡಿ ribo. That means ma'am, here this deoxyribonucleic acid is here, ribonucleic acid. That means maybe whatever is absent here might be present here. So presence of the oxy group or the oxygen group. So this is the main understanding of it. Okay, now when I told that these two nucleic acid, students, DNA ಮತ್ತೆ RNA, ಇವಾಗ ನೀವು ಗೊತ್ತಿರಬೇಕಲ್ಲ ಮ್ಯಾಮ್, DNA ಎಲ್ಲ ಆರ್ಗ್ಯಾನಿಸಮ್ದು ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್, RNA ಎಲ್ಲ ಆರ್ಗ್ಯಾನಿಸಮ್ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಅಂತ ಡೌಟ್ ಬರುತ್ತೆ ಅಲ್ವಾ. ಬಟ್ ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಲ್ಲಿ ನಮ್ಮ DNA ಬಂದ್ಬಿಟ್ಟು, ಓಕೆ, DNA is a genetic material for most of the organism. Okay, it is a genetic material for most of the organism. All organism anta baribedi, most of the organism anta you can write. Okay, done? Okay, now next illi nodi, what about RNA ma'am? Students, RNA is a genetic material. RNA is a genetic material in virus. It's a genetic material in what? It is a genetic material in virus, like your tobacco mosaic virus, TMV virus. Okay, tobacco mosaic virus, like your TMV virus. Okay, so this is clear? Yes or no? Understood? Super duper understood? Genetic material for most of the organisms, RNA genetic material in virus. Okay, TMV. Clear students? Okay. ಇವಾಗ Now you understood that, okay, this is there, this fellow is there, most of the organism. But do you think the absence of oxygen ilva? Ma'am, that might be a trouble, ma'am, later, because something is absent in our life. Let's say, you know, let's say in my life, students, one Rolls Royce is absent. Avun dukka tumba kashta. It's something that I'm very sad about, that I don't have a Rolls Royce in my life. So like that in your life also, there might be something that you don't have. And maybe your friend has it, you do not have it, or your cousins has it, you do not have it. Maybe you don't have a house, or you don't have an iPhone, or you don't have something. So something is absent means, ma'am, then definitely problem will happen. No, ma'am. He'll also be sad. Who will be sad? DNA will be sad. But that is not the case here. Because of that absence, okay, listen to me carefully. Because of this absence, students, it makes this fellow more structurally and chemically stable. Understood? It makes them more structurally and more chemically stable. Understood? We'll be thinking, no, oh, one absence means obviously, ma'am. Okay, FF Gamer is telling, no, ma'am, absence will make the stability. Very good. Structural and chemically, it will make it more stable because of this absence. But at the same time, because of the presence, this fellow will become highly reactive. Now, just imagine, I have Rolls Royce. Just imagine. Okay. Let's say someone is telling me, take one NEET class. Take one Mission MBBS class. Gopika Ma'am, please take one Mission MBBS class. I'll be like, no, I'm too rich, yaar. I don't want to take class for students. I don't want to take it for free. Make my batch. Okay, make my batch. Let the batch be for like, let's say 80,000 rupees. Now, because why? Because I have the presence. I have presence of money. So that's why I'm like very reactive. Okay, I'm going to say no to everyone. This is just an example. Okay, keep it as an example to make you remember this. That presence of oxygen makes this fellow highly reactive, and absence of this makes him structurally and chemically stable. Clear? Done, done, done. Awesome. Okay, this is just the introduction, students. Nucleic acid enide, just the introduction. Now you will be thinking, ma'am, RNA is a genetic material only for your tobacco mosaic virus. Aur bere eno kelsa ilva, ma'am. Ashtena, ma'am, kelsa just on genetic material for virus. Illa, students. Aurge bere salpa kelsa ide. Yen kelsa, ma'am. So they have other job, like they will act as adapter. Okay, they will act as adapter. They will act as catalyst. Okay, that increases the rate of the reaction. Then students, very main important thing, they act as messengers. Remember now Facebook start mada time alli messenger anta ಒಂದು app ಇತ್ತು. So messenger alli, this chapter, because this chapter is the highest weightage chapter, PS, highest weightage, 40 marks weightage. So we thought first we will start high weightage chapters, because already sexual reproduction flowering plant and madidini channel alli. So I've done it multiple times. For FA1 madidini, KC thing madidini, so I didn't want to repeat. So adike molecular basis of inheritance, and also high weightage chapter nimige first day on the motivation 40 marks al nanage at least I'm able to solve this much anta. Okay, now nucleic acid. See, when I keep saying about nucleic acid, you will be thinking, ma'am, please talk about the structure also. Why you only telling about the, you know, just about it? Okay. I didn't get you. What of basic andre? Okay. So here, structure of nucleic acid. Students, illi, first of all, let's understand who gave this nucleic acid word or what happened. Idu NEET level al tumba important. So actually the founders, okay, founders anta heltivi alva. The founders of these people are going to be one person called as Frederick Mischer. Okay, Frederick Mischer. He is the one, okay, Frederick Mischer was the one who was working on pus cells. Okay, who was working on pus cells, okay. And students, he found out there's something, there is one cell that has all the information, and he called that cell, or he called that component as nuclein. What did he do? He called that component as nuclein. Now, nuclein understood, ma'am, but how did this word nucleic acid come in? So, students, this is NEET level important. So, the word nuclein came from a scientist called as Altman. Okay, Altman came and he was like, wait, this nuclein has acidic property. This nuclein has what? It has acidic property. So, we will call this nuclein as nucleic acid. Makes sense, alva? Okay? Yes. See, this chapter basics andre biomolecules. Nim 11th alli chapters. So, ee chapter alli nan foundation 0 inda start martini. Nimigella artha agutte. Because sexual reproduction, flowering plants and each chapter connection enu illa. But biomolecules, principles of inheritance and molecular basis of inheritance ige ಸ್ವಲ್ಪ connection ide. Okay. So, our chapters, principles of inheritance ಮತ್ತೆ ಇದು simultaneously we will be doing. Okay. So, ನಿಮಗೆ ಆ doubt ಬರಲ್ಲ ಅಲ್ಲೋ ಅಯ್ಯೋ ಮ್ಯಾಮ್ ನನಗೆ ಅದು genetics ಏನು ಗೊತ್ತಿಲ್ಲ. I don't know what is monohybrid, dihybrid cross. Now, here what to do. So, biomolecules you will have to do. Okay. Biomolecules that we will teach you. So, look here. Altman found out that this nuclein has acidic property, and because of that, students, he started calling this nuclein as nucleic acid. He started calling this what? Nucleic acid. Understood? He started calling this what? Nucleic acid. Done. Artha aita? So, this nucleic acid is what? Again, your DNA and your RNA. DNA and your RNA. Done. Awesome. Okay. Now, let's understand the structure of this nucleic acid. What is the structure of this nucleic acid? What is this fellow made up of? So, this nucleic acid, students, nucleic acid is actually made up of, okay, nucleic acid is made up of nucleotides. Made up of what? Nucleotides. Nucleotides. What is this nucleotides? I'll tell you. But understand, this nucleic acid, now let's say your double-stranded DNA, ilva? This double-stranded DNA, ilva? So, this double-stranded DNA is made up of what component? Okay, what component? So, here you go and you will start. You will notice that like this, many individual molecules are there. Okay, many individual molecules are there. That is called as the nucleotide. Okay, that is called as the nucleotide. So, these DNA molecules, individual units eno nucleotides. Individual units eno nucleotides. Okay. So, these nucleotides, students, they are made up of more components. Okay, they are made up of 3 components. First one, it is called as the sugar. Okay, first component is called as the sugar. Next component is called as the nitrogenous bases. Nitrogenous bases. And the third component is called as the phosphate group. Phosphate group. Artha aita ellargu? So, very simple. Okay, let's say, yes, don't worry dear, we will start from basics, like whichever chapters, whichever you have a problem, please let us know in the comments. Nimage already gotu comments ella ನಾವು ಓದ್ತೀವಿ. ಅದರಿಂದ ಏನಾದರೂ ನಿಮಗೆ ಪ್ರಾಬ್ಲಮ್ ಇದ್ರೆ we definitely do whatever is required for you. But ನಿಮಗೆ ನೀಟಿಗೆ ಕ್ವಾಲಿಟಿ ಕಂಟೆಂಟ್ ಕೊಡಬೇಕಂತ we are doing this. Okay, ಇಲ್ಲ ಅಂದ್ರೆ ಈ ಚಾನಲ್ KCT ಮಾತ್ರ we would have done. But then we noticed that many students are there who wants NEET. ತುಂಬಾ ಕಮೆಂಟ್ಸ್ ಬರ್ತಾ ಇತ್ತು. So, ಅದಕ್ಕೆ ಮಾಡ್ತಾ ಇದೀವಿ. So, I want all of you to share this video with all your friends and learn as much as possible. Okay. So, ಇಲ್ಲಿ ನೋಡಿ students, sugar, phosphate group ಮತ್ತೆ nitrogenous base. ಈ 3 components ಒಟ್ಟಿಗೆ ಸೇರಿ nucleotide formation ಆಗುತ್ತೆ. So, let's say ನಮಗೊಂದು sugar molecule ಇದೆ. Okay, let's just draw one shape. One sugar molecule is there. Like this. Okay. Okay. Sugar on the 5th position here, you will have a phosphate group. Okay, let's draw one triangle. Okay, there's a phosphate group. Phosphate group. And from the first carbon, let's say there will be a nitrogenous base. Nitrogenous base. So, each on combination, one nucleotide formation will happen. one nucleotide formation will happen. Clear to all of you? So this is one nucleotide. One nucleotide. One nucleotide. Done, done, done. Okay. Now, so introduction is clear. So what did I teach you? I told you Friedrich Mischer was the person when he was working on this pus cells. Okay. So students, example andre, you fridge open mana time ali, let's say by mistake, let's say you are searching for chicken. Okay, you are searching for chicken biryani in the fridge. You never know where chicken biryani is kept. But let's say you fridge open madbittu one box etkondidivi. Sudden aga box olagade biryani irutte. So andre eno by fluke, by luck ma'am nanage siktanta. So aa tarane illi by fluke illa andre by luck Friedrich Mischer found that there was a cell which had this information, something that was different, so he called that cell as nuclein. Then another scientist came called Altman, in the NEET point of view important, Altman, who told that wait, this nuclein has some acidic properties, so why don't we call it nucleic acid? And that is the two nucleic acid, DNA matte RNA, deoxyribonucleic acid matte ribonucleic acid. Now, this nucleic acid, what is it made up of? It is made up of individual components called as nucleotides. And what is this nucleotides made up of? Nucleotides are made up of three components. Another students, nitrogenous base, pentose sugar and phosphate group. So you can remember like this, NPP. Okay? NPP. That means nitrogenous base, pentose sugar and phosphate group. Understood? Nucleoside I'll come into 1 second. So, ee tara nimage DNA structure individual ag open ಮಾಡಿ ನೋಡಿದ್ರೆ ನಿಮಗೆ ನೋಡಬಹುದು nucleotides ಇರುತ್ತೆ. Now each nucleotides is going to have a sugar, phosphate and a nitrogenous base. Done, done, done. Okay, now we are going to study about nucleosides and nucleotides, but before that students, you have a very important box in your textbook, that is this box. Okay, they have given you a certain set of organisms and their number of DNA molecules or number of nucleotides or number of base pairing. Now what is base pairing andre? Now DNA strands ee tara iruttalva, like this DNA strands will be one strand, one more strand. They are going to be complementary, but even though they are complementary, there should be something that keeps them together like this, to hold them together like this. Yes or no? To hold them together students, we are going to have something called as the base pairs. Okay, base pairs. So the base pairs, again I'll teach you in detail, but right now here. Okay, look here. So 5174 has 5386 nucleotides. I hope all of you are smashing the like button. Attendance mark ಮಾಡಿ miss ಮಾಡಬೇಡಿ. Okay, ಈ numbers ಎಲ್ಲ ನಿಮಗೆ by heart ಆಗಿ ಗೊತ್ತಿರಬೇಕು. 5386 nucleotides. So students, ಇಲ್ಲಿ nucleotides mention ಮಾಡಿದ್ದಾರೆ. That means it is a single stranded structure. Okay, single stranded structure, because only nucleotides are mentioned. Now you see bacteriophage lamla 48502 base pair. 48502 base pair. That means these people are going to be paired. Okay, double stranded there. E. coli 4.6 into 10 to the power 6. Okay, so here it is copy pasting agide. 4.6 into 10 to the power 6 base pairs. Okay, now haploid content of human, important NEET point of view. ನಾನು ಕ್ಲಾಸಲ್ಲಿ ಹೇಳ್ತೀನಿ ನೀಟಿಗೆ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂದರೆ ಪ್ಲೀಸ್ ಮಾರ್ಕ್ ಮಾಡಿ NCERT ಅಲ್ಲಿ. ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಕಂಟೆಂಟ್ ಆಫ್ ಹ್ಯೂಮನ್ ಸ್ಟೂಡೆಂಟ್ಸ್ 3.3 into 10 to the power 9 ಬೇಸ್ ಪೇರ್. ಸೊ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ಸ್ ಇದೆ ಅಲ್ವಾ, ಸೊ ದಿಸ್ ಇಸ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಸೆಲ್. ಸೊ ಡಿಪ್ಲಾಯ್ಡ್ ವಿಲ್ ಬಿ ವಾಟ್? Diploid will be what? 3.3 into 10 to the power 9 into 2 you have to do. Okay, so here it will be 6.6 into 10 to the power 9 base pair. That will be your haploid content, and this is NEET point of view, very important. NEET point of view, very, very important. Clear? Yes? Okay. Now, nitrogenous bases. Before I go to nitrogenous bases, okay, I'll do nitrogenous bases. I'll do this nucleoside and nucleotide. Then I'll come to nitrogenous bases. Okay. Okay. Now nucleoside. Someone was asking, no ma'am, what is nucleoside? I explain ಮಾಡ್ತೀನಿ. Okay. NCERT lines ಇಂದಾನು explain ಮಾಡ್ತೀನಿ. So ಇಲ್ಲಿ ನೋಡಿ students, nucleoside ಮತ್ತೆ nucleotide. Okay. What is this nucleoside and nucleotide ನೋಡೋಣ. Now students, very simple. ನಮಗೆ nucleotide ಬಗ್ಗೆ ಗೊತ್ತು already ಅಲ್ವಾ. nucleotide means what? Sugar is there, okay, pentose sugar, okay, nitrogenous bases and phosphate group, okay, phosphate group, correct? This is already understood that nucleotide, we have pentose sugar, nitrogen base and phosphate group. Now they are talking about nucleoside. Now let's take example, okay, all of you are studying, okay, all of you are studying, and let's say all of you have become very close friends, but there is this one student who is disturbing the class, okay, one student who is disturbing the class. Okay, one student who is disturbing the class. Now we'll push that student to one side. What we will do? We'll push that student to one side. So aa tarane illi enagutte students, when you push one person to one side, that is called a nucleoside. ಇವಾಗ ನೀವೆಲ್ಲ ಫ್ರೆಂಡ್ಸ್ ಆಗಿದ್ದೀರಾ, ಬಟ್ ಒನ್ ಸ್ಟೂಡೆಂಟ್ ಕಂಟಿನ್ಯೂಸ್ಲಿ ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ, ನಾನ್ ಸ್ಟಾಪ್ ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ, ಯು ಆರ್ ಗೆಟಿಂಗ್ ಡಿಸ್ಟರ್ಬ್. ಸೊ ಅವ್ರನ್ನ ನೀವೇನು ಹೇಳ್ತೀವಿ, ಅಲ್ಲಿ ಸೈಡಲ್ಲಿ ಹೋಗಿ ಕುತ್ಕೋ ಪ್ಲೀಸ್, ನನ್ನ ಡಿಸ್ಟರ್ಬ್ ಮಾಡಬೇಡ ಅಂತ ಹೇಳ್ತೀರಾ, ಓಕೆ, ನಾರ್ಮಲ್. ಸೊ ವೆನ್ ಯು ಪುಷ್ ದಟ್ ಒನ್ ಪರ್ಸನ್ ಟು ದ ಸೈಡ್, ಇಟ್ ವಿಲ್ ಬಿಕಮ್ ಅ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್. ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಲ್ಲಿ ಪೆಂಟೋಸ್ ಶುಗರ್, ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಆರ್ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್ ಇಸ್ there. Now if I push this person, if I push this person to the side, okay, I am pushing this person to a side, then I'll get what here? I am going to get a nucleoside. A nucleoside. Is this clear? Is this clear? Yes or no? So pentose sugar and nitrogen base together will form your nucleoside because I pushed phosphate to one side. Okay, when I pushed phosphate to one side, what happened? It led to formation of nucleoside. It led to formation of nucleoside. Now students, diagram tumba important nimige. NEET point of view diagram tumba important. Okay, so namak first nucleoside draw madana. So normally nam sugar group bandbit 5 carbon sugar. Okay, so biology teachers, chemistry, okay, yes, almost good diagram. Okay, ಇಲ್ಲಿ ನೋಡಿ, ಇವಾಗ ಇಲ್ಲೊಂದು ಮೇಜರ್ ಡಿಫರೆನ್ಸ್ ತೋರಿಸ್ತೀನಿ. Okay, okay, done. So now look here students, let's say this is RNA molecule and this is DNA molecule. ಇವಾಗ ನಮಗೆ ಕಾರ್ಬನ್ ಕೌಂಟ್ ಮಾಡೋಣ. ಓಕೆ, 1 ಕಾರ್ಬನ್, 2 ಕಾರ್ಬನ್, 3 ಕಾರ್ಬನ್, 4 ಕಾರ್ಬನ್ ಆ್ಯಂಡ್ ಇಲ್ಲಿ 5th ಕಾರ್ಬನ್ ಬರುತ್ತೆ, CH2OH ಅಂತ ಬರುತ್ತೆ. ಓಕೆ, ಚಿಕ್ಕದಾಗಿ ಬರೀತೀನಿ ಬಿಕಾಸ್ ಐ ಡೋಂಟ್ ವಾಂಟ್ ಯು ಪೀಪಲ್ ಟು ಕನ್ಫ್ಯೂಸ್. ಲಿಸನ್ ಟು ಮೀ ಕೇರ್ಫುಲಿ, ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್ ಕಾನ್ಸೆಪ್ಟ್. ಓಕೆ, ಸೊ 1, 2, 3, 4, 5th ಕಾರ್ಬನ್, 5 ಕಾರ್ಬನ್ ಮಾಲಿಕ್ಯೂಲ್, ಪೆಂಟೋಸ್ ಶುಗರ್ ಇದು. ಅದೇ ಥರ ಡಿ ಎನ್ ಎ ಅಲ್ಲೂ ಇರುತ್ತೆ. ಓಕೆ, ಸೇಮ್ ಥಿಂಗ್ ಇನ್ ಡಿ ಎನ್ ಎ ಆಲ್ಸೋ, 1 ಕಾರ್ಬನ್, 2 ಕಾರ್ಬನ್, 3 ಕಾರ್ಬನ್, 4 ಕಾರ್ಬನ್ and then you have CH2OH. okay, CH2OH, that becomes the 5th carbon. ಇವಾಗ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಈ ಸೆಕೆಂಡ್ ಕಾರ್ಬನ್ ಫೋಕಸ್ ಮಾಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ಈ ಸೆಕೆಂಡ್ ಕಾರ್ಬನ್ ಅಲ್ಲಿ ಈ RNA ಗ್ರೂಪ್ ಅಲ್ಲಿ, ಓಕೆ, ಸೊ ನಾನು ಇಫ್ ಯು ವಾಂಟ್ ಐ ರೈಟ್ ದ ಹೋಲ್ ಥಿಂಗ್. ಓಕೆ, so now let's say if I write only this much, okay, will it confuse you? No, right? If I write only this much, will it confuse you? Okay, just look at this. This is the structure. Okay, ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಕರೆಕ್ಟ್ ಆಗಿ ನೋಡಿ, ಇಲ್ಲಿ here second carbon, can you see one OH is there? Can you see the second carbon, there's one OH? Yes ma'am, I can see. Now let me draw the DNA structure. Okay, DNA structure also I'm drawing. OH, H, 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 OH, H and H. Okay, now look at this. In the second carbon, okay, in the second carbon, RNA has an OH, okay, RNA, okay, second carbon has an OH and here second carbon has a hydrogen group, a H group. correct? Understood? So ನಾನು ಆಲ್ರೆಡಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ ದಟ್ ರೈಬೋನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಂದರೆ ದೇ ಹ್ಯಾವ್ ಎನ್ ಆಕ್ಸಿಜನ್ ಗ್ರೂಪ್ ಎಕ್ಸ್ಟ್ರಾ ಆ್ಯಂಡ್ ಡಿಆಕ್ಸಿ ರೈಬೋನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಂದರೆ ಡಿಆಕ್ಸಿ ಅಂದರೆ ಒಂದು ಆಕ್ಸಿಜನ್ ಕಮ್ಮಿ ಆಗಿರುತ್ತೆ. ಸೊ ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಇಲ್ಲಿ ಆಕ್ಸಿಜನ್ ಒಂದು ಜಾಸ್ತಿ ಇದೆ, ದಟ್ ಇಸ್ RNA ಮಾಲಿಕ್ಯೂಲಲ್ಲಿ ಇಲ್ಲೊಂದು ಆಕ್ಸಿಜನ್ ಇದೆ, ಅದೇ ಆಕ್ಸಿಜನ್ ಇಲ್ಲಿ ಆಬ್ಸೆಂಟ್ ಆಗಿದೆ. ಸೊ 2-H ಅಲ್ಲಿ 2-H and 2-OH. So this is the structure of your sugar. Sugar is clear to all of you? Yes. Now students, just because this is there, we cannot just let it go like this. You have to understand that your DNA molecules, okay, DNA molecules because of this becomes less reactive, okay, less reactive and more stable, okay, more stable, less reactive, more stable. Okay, done. Okay, done. Less reactive. Yes, less reactive, more stable. Now, presence of this. Okay, presence of this led to what? It led to two things. More reactive. More reactive. Less stable. More reactive, less stable. Yes? Correct? Which carbon? Third carbon interchange. Artha agilla dear? Correct agide third carbon. So illi nodi 1-, 2-, 3-, 4-. ಈ 3-OH ಬಂದ್ಬಿಟ್ಟು ಬಾಂಡಿಂಗ್ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಓಕೆ, 3-OH ಬಂದ್ಬಿಟ್ಟು ಬಾಂಡಿಂಗ್ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ ಲೇಟರ್. ಓಕೆ, ಲೇಟರ್. Is it a doubt or what? You like explain me little so I understand. Okay. Done. Okay, now this is understood. We understood what is a nucleo. Now we have understood a pentose sugar. Now next we have to understand a nitrogen base. Adamele we should understand a phosphate group. Alva. So illi nodi. If you want, we will duplicate this. Okay. Illi nodi. Pentose sugar you have understood. Now from the pentose sugar you have to understand a nitrogenous base. Okay. So nitrogenous base. Okay. 1 minute. Okay, illi nitrogenous base. Nitrogenous base students illi artha madkoli. What is nitrogenous base? You have purine and you have pyrimidine. You have purine and you have pyrimidine. These are the two nitrogenous bases. Okay, these are the two nitrogenous bases. Okay, purine matte pyrimidine. Ivaga one trick kodtini, correct ag try to listen. So students, illi remember pure as gold. Okay, my Karnataka students' heart is pure as gold. Okay, amele remember cut, C-U-T, cut. Okay, C-U-T, cut. Now what is this pure as gold? Nodi, pure is for the purine. Okay, pure is for the purine. Clear? Yes? Awesome. Okay, and then as gold, ilva. For that you write adenine and guanine. Adenine and guanine. Okay, pure as gold. Adamele illi nodi one nimisha ee wire pyrimidine ge remember cut. Okay, C-U-T, cut. Okay, C for cytosine, U for uracil, okay, T for thymine. Okay, thymine. So pyrimidine, C-U-T, cut. Ellaru heli chat box alli pure as gold and C-U-T, cut. So nitrogenous base are two types, purines and pyrimidines. Under purines you have adenine and guanine, and under pyrimidines you have cytosine, uracil and thymine. You will be thinking, ma'am, illi 2 ide, illi 3 ide, ma'am, yen ma'am. So illi nodi students, Why does it have 2 and 3 and all of that? ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಅರ್ಥ ಆಗುತ್ತೆ. Adenine is going to be there in DNA and RNA. Okay. ಇಲ್ಲಿ ನೋಡಿ guanine also DNA and RNA. Okay. So both are there in DNA and RNA. This question has come in the NEET. Okay. So okay, NEET PYQ. So RNA, DNA. ಇಲ್ಲಿ ನೋಡಿ cytosine. Cytosine also DNA and RNA. Yes or no? ಇಲ್ಲಿ ನೋಡಿ ಯುರಾಸಿಲ್ ಬಂದ್ಬಿಟ್ಟು ಸ್ವಲ್ಪ ಸ್ಪೆಷಲ್ ಯುರಾಸಿಲ್ ಬಂದ್ಬಿಟ್ಟು ಮ್ಯಾಮ್ ನನಗೆ DNA ಬೇಡ ಮ್ಯಾಮ್, ನಾನು RNA ಗೆ ಹೋಗ್ತೀನಿ. ಸೊ ಹಿಯರ್ he is only going to be present in RNA. So you will be thinking, okay, then who is going to be in DNA? DNA we will have thymine. So to be specific students, thymine in DNA will be replaced by uracil in RNA. Now to make you understand this, I will show you here. Okay, let's say this pink color. ಸ್ವಲ್ಪ thick draw ಮಾಡ್ತೀನಿ. Is this clear to all of you? ಅರ್ಥ ಆಯ್ತಾ? ಇಷ್ಟು ಕಾಂಪ್ಲಿಕೇಶನ್ ಇದೆಯಾ ಮ್ಯಾಮ್ ಜೆನೆಟಿಕ್ಸ್ಗೆ? ಅಷ್ಟು ಕಾಂಪ್ಲಿಕೇಶನ್ ಇಲ್ಲ ಸ್ಟೂಡೆಂಟ್ಸ್. ಇಟ್ಸ್ ಈಸಿ. ಓಕೆ, ಇಫ್ ಯು ಲಿಸನ್ ಪ್ರಾಪರ್ಲಿ ಇಟ್ಸ್ ಈಸಿ. ಲೆಟ್ಸ್ ಸೇ ದಿಸ್ ಇಸ್ ಯೋರ್ DNA. ಓಕೆ, ಇದು DNA. ಓಕೆ, ಇದು RNA. ಏನು ಹೇಳಿದೆ? ಥೈಮಿನ್ ವಿಲ್ ಬಿ ರಿಪ್ಲೇಸ್ಡ್ ಬೈ ಯುರಾಸಿಲ್ ಅಂತ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಅಡೆನೈನ್, guanine, cytosine, thymine, adenine, uracil, thymine, guanine, cytosine. ಈ ತರ ಒಂದು I have just written all the nitrogenous bases. Okay, ನಾವು ಇಲ್ಲಿ ನೋಡಿ RNA ಬಂದ್ಬಿಟ್ಟು ಏನು ಫಾರ್ಮ್ ಆಗುತ್ತೆ ಇಲ್ಲಿ? RNA ಒಂದು ನಿಮಿಷ. RNA ನೋಡಿ A will bind with whom students? A will bind with, let's say, now you don't have to worry about it, let's say, but you know, let's say A will bind with uracil, right? So U. Instead of that, let's not write complementary, let's just write the same thing here. So here A will be there, okay, here G will be there, here C will be there, here T will be replaced by U. T will be replaced by U. Again, let's say A, again T will be replaced by U, G will be G, C will be C. So this becomes a RNA. Understood? So wherever thymine is there, thymine is replaced by uracil. Understood? Thymine is replaced by uracil. Done? Yes. Madesha understood? All of you understood? Attendance mark ಮಾಡಿದ್ದೀರಾ? Yes or no? Attendance done? Let me see your attendance. Wait. I'll see how many loyal students are there in the class. Yes, quickly mark the attendance. Okay? Awesome. Good going, good going. Okay? All of you are marking the attendance. Awesome. Clear. Done. So this is clear. Any doubts here? ಸ್ವಲ್ಪ ಇಂಪಾರ್ಟೆಂಟ್ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ನೋಡಿ. ಪ್ಯೂರಿನ್ ಬಂದ್ಬಿಟ್ಟು ಅಡಿನೈನ್ ಅಂಡ್ ಗ್ವಾನಿನ್, ಪಿರಮಿಡಿನ್ ಬಂದ್ಬಿಟ್ಟು ಸೈಟೋಸಿನ್, ಯುರಾಸಿಲ್ ಅಂಡ್ ಥೈಮಿನ್. ಓಕೆ, ಸೊ DNA and RNA ಮತ್ತೆ DNA it will be present. This is a NEET PYQ, so please remember it. Okay, ಇಲ್ಲಿ ನೋಡಿದ್ರೆ pure as gold is a trick to remember all the purines. Okay, it is a trick to remember all the purines. Okay, and here the trick to remember pyrimidines is here. Okay, pyrimidines is here. Then DNA, RNA. Thymine will be replaced by uracil. Thymine will be replaced by uracil. Okay? Yes. Done, ma'am. Okay, now. Okay. Look here, students, the structure. So, okay, one thing. Okay, thank you, Rajesh. Hi, Anita. So, students, ಅದೇನು ಕನ್ನಡದಲ್ಲಿ ಒಂದು ಪ್ರಾವರ್ಬ್ ಅಲ್ವಾ, ಹೆಸರು ಚಿಕ್ಕದು ಕೀರ್ತಿ ದೊಡ್ಡದು, something like that they say in Kannada, no? Just like that, purine is a small name, P-U-R-I-N-E, ma'am. Pyrimidine, big name, P-Y-R-I-M-I-D-I-N-E, pyrimidine. So ಆ ಥರನೇ ಇಲ್ಲಿ purine is a 9-membered ring. ನೇಮ್ ಚಿಕ್ಕದು ಬಟ್ ಸ್ಟ್ರಕ್ಚರ್ ದೊಡ್ಡದು. ಓಕೆ, ಪ್ಯೂರಿನ್ is, look at the structure, it's a 9 member. So ಇಲ್ಲಿ ನಿಮಗೆ NEET point of view ಸ್ವಲ್ಪ extra information. 9 membered ring. Again, you'll study this in biomolecules. Since you people didn't study that, I'm teaching you here only. It's a 9 membered ring. Okay, ಆಮೇಲೆ you have to know the places where the nitrogen groups are going to be present. ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಸ್ಟೂಡೆಂಟ್ಸ್ ಇಲ್ಲಿ ಕರೆಕ್ಟ್ ಆಗಿ ನೋಡ್ಬೋದು where the nitrogens are present. 1 2 3 4 nitrogens are there. So nitrogen groups are present in the 1 3 7 9th positions. Okay, so nitrogen groups are present in 1 3 7 9th position. Ha, Murti chikkadaದರೂ ಕೀರ್ತಿ ದೊಡ್ಡದು. Yes, correct. Did I say ulta? I'm sorry if I told ulta, but then I've heard it. Okay, I've heard it. So yes, 1 3 7 9 students, you have a nitrogen position. Okay, N group is going to be present. Okay, now 9 membered ring, you understand 2 rings are there. So ಇಲ್ಲಿ ನೋಡಬಹುದು 2 rings ಇದೆ. So you can see, okay, 1 2 3 4 5 6 7 8 9. So like that 9 positions are there. Cyclic and non-cyclic labelings you have to do. ಇಲ್ಲಿ ನೋಡ್ರಿ pyrimidine, name is bigger. Okay, hi Rajesh, name is bigger, but here structure is only 6 membered. 6 membered structure, 6 membered ring. And students, the nitrogen position is only at 2 places. What is it? 1 3 N positions are there. 1 3 N positions are there. Understood? Yes, this is a little additional, but it is required for you. Adenine and guanine, thymine, cytosine and uracil. Okay, now keep this in mind, the structure, because here we are going to study about bonding. Okay, we are going to study about bonding. Phosphate bagay, there's nothing more about phosphate. Okay, it's just P double bond O, O and O. Okay, that's all is there. Now we are going to study about what? We are going to study about your groups, I mean bonding. Okay, so if I have to form a nucleotide, or let's say nucleoside also, I need to make bond. Ilva, 3 ಜನ ಮಧ್ಯದಲ್ಲಿ ಒಂದು ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಇರಬೇಕು, ಇಲ್ಲ ಅಂದ್ರೆ they will not work together. ಅಲ್ವಾ? So let's say I want to work, Baswa sir should work and Catherine ma'am should work. We are the 3 people. Okay, we together make the, let's say, Vedantu Karnataka. ಇವಾಗ ನಾವು 3 ಜನ ವರ್ಕ್ ಮಾಡಬೇಕಂದ್ರೆ ನಮ್ಮ ಮಧ್ಯದಲ್ಲಿ ಒಂದು ಬಾಂಡ್ ಇರಬೇಕು. ಓಕೆ ಸರ್, ನೀವು ಆ ವಿಡಿಯೋ ಮಾಡಿ ಚೆನ್ನಾಗಿರುತ್ತೆ, ನೀವು ಈ ಕ್ಲಾಸ್ ಎತ್ಕೊಳ್ಳಿ ಚೆನ್ನಾಗಿರುತ್ತೆ, ಆ ಥರ ಒಂದು ಬಾಂಡ್ ಇರಬೇಕು. ಆ ಥರನೇ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಮಧ್ಯದಲ್ಲೂ ಅಂದರೆ ಶುಗರ್, ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್, ಫಾಸ್ಫೇಟ್ ಮಧ್ಯದಲ್ಲೂ ಒಂದು ಬಾಂಡ್ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ಆ ಬಾಂಡ್ ಬಗ್ಗೆ ಇಲ್ಲಿ ಲಿಸನ್ ಕೇರ್ಫುಲಿ. ಓಕೆ, ಇಟ್ಸ್ ವೆರಿ ಸಿಂಪಲ್. ಸೊ ಫಸ್ಟ್ ನಾವು ಸ್ಟ್ರಕ್ಚರ್ ಡ್ರಾ ಮಾಡೋಣ ಸ್ಟೂಡೆಂಟ್ಸ್. So structure one bit, first you are going to draw a sugar molecule. Okay, now you know to draw a sugar molecule, I hope. I'll draw it a little more bigger. Okay, done. Sugar molecule done. You want to put the lines, you can put the lines as well. So here you are going to have the 5th carbon, that is CH2OH. Okay, now first what did I tell you? Nucleoside andre eno? Nucleoside andre when I push phosphate to one side, alva. So let's say this nitrogen base is going to make bond with this sugar is going to make bond with nitrogen base, alva. So first I have to draw the nitrogen base. So we will draw a purine. Okay, let's say adenine or guanine. Okay, this is the structure. Get it, ring ide students, 9 positions are there. Okay, now let's say this is the 9th position, this is the 9th position. So whenever a sugar, remember this, whenever a sugar bonds with a nitrogen base, they're always going to bond in the 1 9th position in a purine. I know this might be little difficult for you to understand, but let's try and understand once again. Okay, let's try and understand once again. What did I say? Okay, what did I say? That is, whenever, okay, whenever ಇಲ್ಲಿ ಬರೀತೀನಿ, whenever a sugar makes a bond with a purine. Okay, purine andre adenine or guanine. Okay, adenine, oh sorry, and alla or adenine or guanine. It will always make a bond, students, from 1 andre first carbon to the 9th position of the purine. Understood? So here 1 dash, here 9. Okay, 9. Yes or no? Understood? Please let me know in the chats. Please give me a thumbs up if all of you understood. Yes. Wait, I can see your chats now. Understood? Okay. So 1 comma 9. Now students, you will be thinking, ma'am, what is the bond that is formed between them? I do not know which bond you are talking about. Yes, the bond that I am talking between them, students, is called as 1 comma 9 glycosidic linkage. What is it called? Glycosidic linkage. 1 dash 9 glycosidic linkage. Understood? So, remember, let's recall it once again. Whenever a sugar, okay, write it in your NCERT and keep. Whenever a sugar bonds with a nitrogen base, not just nitrogen base, let's be specific. Whenever it bonds with a purine, okay, it will bond in the 1 comma 9th position. Andre, first carbon of the sugar, 9th position on the purine. Okay. Whenever the sugar bonds with a pyrimidine, students, avaga illi enagutte 1 comma 6th position. 1 comma 6th position. Andre pyrimidine 9 positions illa, irade 6 carbon alva, sorry 6 positions alva, 6 carbon. So adarli sugar do first molecule and pyrimidine 6th position. Okay, 6th position. So ille nodi diagram il nodbodalla. So whenever they are binding, okay, one will bind here, sorry, okay, one will bind to the 9th position, one will bind to the 6th position. Understood? That is what they are trying to say here. Okay, this is all very important from NEET point of view. Now, this two, okay, sugar plus purine illandre sugar plus nitrogenous base, okay, il nitrogenous base anta baritini, NB. Nitrogen base, together what do you call it? Ma'am, together I am going to call it, this much is going to be called a nucleoside. ಏನು ಕರೀತೀರಾ? ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಅಂತ ಕರೀತೀರಾ. ಓಕೆ, ಹಾಯ್ ರುದ್ರ, ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಅಂತ ಕರೀತೀರಾ. ಇವಾಗ ಮ್ಯಾಮ್ ಮ್ಯಾಮ್ ಫಾಸ್ಫೇಟೇ ನೋಡಿಲ್ಲ ಮ್ಯಾಮ್, ವೇರ್ ಇಸ್ ಫಾಸ್ಫೇಟ್ ಗೋಯಿಂಗ್ ಟು ಕಮ್ ಅಂಡ್ ಅಟ್ಯಾಚ್? ಹಿಯರ್ ಕಮ್ಸ್ ದ ಆನ್ಸರ್ ಫಾರ್ ಇಟ್. ಫಾಸ್ಫೇಟ್ ಬಂದ್ಬಿಟ್ಟು ಸ್ಟೂಡೆಂಟ್ಸ್ ಈ OH ಇಲ್ವಾ, 5th ಕಾರ್ಬನ್ ಅಲ್ಲಿ ಬಂದು ಅಟ್ಯಾಚ್ ಮಾಡ್ತಾರೆ. ಅಂದ್ರೆ ಇಟ್ ವಿಲ್ ರಿಮೂವ್ ದಿಸ್ ಹೈಡ್ರೋಜನ್ and it will make a bond here. Okay, P double bond, ಕೆಳಗೂ ಬರಿಬೋದು. Like here you don't have to put double bond, ಇಲ್ಲ ಅಂದ್ರೆ ಕೆಳಗೂ ಬರಿಬೋದು, it's up to you. So O, you O, so here like this. Okay, they are going to make a bond in such a way. Now remember that I told color difference. Okay, oh that's awesome Rudra, thank you so much. So ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇಲ್ಲೇನಾಗುತ್ತೆ ಒಂದು ಶುಗರ್ ಹ್ಯಾಸ್ ಟು ಬೈಂಡ್ ವಿತ್ ಅ ಫಾಸ್ಫೇಟ್. ಸೊ ಇಷ್ಟು ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್. ಇವಾಗ ಇದು ಕಂಪ್ಲೀಟ್ ಆಗಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಆಯ್ತಾ ಅಲ್ವಾ, ಬಿಕಾಸ್ ಫಾಸ್ಫೇಟ್ ಇದೆ, ಶುಗರ್ ಮಾಲಿಕ್ಯೂಲ್ ಇದೆ, ಮತ್ತೆ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್ ಇದೆ. ಇವಾಗ ಶುಗರ್ ಮತ್ತು ಫಾಸ್ಫೇಟ್ ಒಂದು ಬಾಂಡ್ ಫಾರ್ಮೇಶನ್, ಇಲ್ಲಾಂದ್ರೆ ಅವರೊಂದು ಕನೆಕ್ಟ್ ಆಗ್ಬೇಕಂದ್ರೆ ಒಂದು ಬಾಂಡ್ ಫಾರ್ಮೇಶನ್ ಖಂಡಿತ. ಆ ಬಾಂಡ್ ಫಾರ್ಮೇಶನ್ ಎಲ್ಲಾಗುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್? ಆ ಬಾಂಡ್ ಫಾರ್ಮೇಶನ್ ಇಲ್ಲಾಗುತ್ತೆ. ಅಂಡ್ ಈ ಬಾಂಡಿಗೆ ನಾವೇನು ಕರಿತೀವಿ? ವಿ ಕಾಲ್ ದಿಸ್ ಬಾಂಡ್, ಓಕೆ, ವಿ ಕಾಲ್ ದಿಸ್ ಬಾಂಡ್, ವಾಟ್ ವಿ ಕಾಲ್ ದಿಸ್ ಬಾಂಡ್? ವಿ ಕಾಲ್ ದಿಸ್ ಬಾಂಡ್ ಆಸ್ ಫಾಸ್ಫೋ. ಸೇಮ್ ಕಲರ್ ಅಲ್ಲಿ ಬರೀತೀನಿ, 1 minute. I'll put it in green so you know that it is bonds. Okay, it's called as phosphoester bond. Phosphoester bond. Artha aita ellarigu? Okay, yes, is this clear? Is this clear? So I'll explain once again in English for all of you. So students, let's say here we have a sugar molecule, here we have a nitrogenous base, here we have a phosphate group. Now I need to make bonding with all these people. Why? Because these three people together form the nucleotide. So if three people are together, I need to tell them, hey, you also hold my hand, you also hold my hand, then only we can together make the nucleic acid. So let's say I am sugar. Okay, I am sugar, and this becomes the nitrogen base. Okay, so I am telling sugar, I am sugar. So he'll be like, okay, fine, I want to bond with you. So let's say this is the purine. Now sugar, this bond is going to be called as 1,9 glycosidic linkage if this is a purine. Let's say this is a pyrimidine. Let's say this is a pyrimidine. It is going to be called what? 1,6, okay, glycosidic linkage. Understood? So pyrimidine 1,6, purine 1,9. Ivaga this much has happened. That means I am a sugar, this is a nitrogenous base. Now I need what? Now I need what? I need a phosphate. Now I'll tell the phosphate to come and bind with me. So phosphate will come. Phosphate came. Phosphate is going to bind where, students? To the fifth carbon. Fifth carbon with a phosphoester bond. So this is going to be a phosphoester bond. Is this clear to all of you? Understood? Artha aita ellargo? Yes. So purine jote combine mada time alli 1,9, okay, 1,9 glycosidic linkage. Pyrimidine jote combine mada time alli 1,6 glycosidic linkage. Phosphate and sugar is going to make a bond called as phosphoester bond. Phosphoester bond. Clear? Okay. After the class, please let me know how the class was, okay? Another changes madbekandre we will do. So this is done. See students, now can I call this whole thing? Can I call this whole thing a nucleotide? Yes ma'am, you can call this whole thing as a nucleotide. So starting from phosphate, having my sugar, then coming here, together I will call it a nucleotide. So bond is clear? Bond is clear? So here students, this is very, very, very important. How many times if I tell it's important also, it's important. Okay, that much important this is. Okay, so remember sugar plus nitrogenous base, if it's a purine, it's going to be 1,9 glycosidic linkage. 1 second, I'll write in more. So sugar plus NB I'm writing, that is nitrogenous base. Let's say if it's a purine, okay, let's say it is a purine, it is going to be what? 1, okay, 9 glycosidic linkage. Okay, glycosidic linkage. Let's say it is a pyrimidine. It's a pyrimidine. Okay, then it is going to be what? It is going to be 1,6, okay, glycosidic linkage. Done? Done, done? Thank you, Chutki. You people have such fancy names. Huh? Chutki, Chinni, huh? All like, I used to watch Chota Beam, students. Chutki. Okay, done? Atta itaido? Everyone understood? Anyone who hasn't understood, please raise your hands. If you understood, you know, put thumbs up in the chat box. Yes. ಅರ್ಥ ಆಯ್ತಾ? ಓಕೆ, ಇಲ್ಲಿ ನೋಡಿ ಇನ್ನೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಟಾಪಿಕ್. ಇಲ್ಲಿ ನಿಮ್ಮ NCERT ಎಲ್ಲ NCERT ಬೇಸೇ, ಬಟ್ ಆದರೆ ಸ್ವಲ್ಪ ಅಡಿಷನಲ್ ಇನ್ಫಾರ್ಮೇಷನ್ ಹೇಳ್ತೀನಿ. ಬಿಕಾಸ್ ನಿಮಗೆ ಗೊತ್ತು NEET 2025 ವಾಸ್ ಲಿಟಲ್ ಡಿಫಿಕಲ್ಟ್, ಸೊ ವಿ ಹ್ಯಾವ್ ಟು ಬಿ ಪ್ರಿಪೇರ್ಡ್ ಫಾರ್ ದ್ಯಾಟ್. ಓಕೆ, ವಿ ಹ್ಯಾವ್ ಟು ಬಿ ಪ್ರಿಪೇರ್ಡ್ ಫಾರ್ ದ್ಯಾಟ್. ಸೊ ಇಲ್ಲಿ ನೋಡಿ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇವಾಗ ಒಂದು ಸಫಿಕ್ಸ್, ಒಂದು ಪ್ರಿಫಿಕ್ಸ್ ಅಂಡ್ ಸಫಿಕ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ ಅಲ್ವಾ. ಸೊ there is one suffix to all of this, that is going to be, if it is a nucleoside. Okay, board is dancing more than me. If it is a nucleoside, okay, the prefix is going to be ine. Okay, ine, I-N-E. Okay, I-N-E, ine. Clear? Yes? Okay. Now, ma'am, ma'am, how does it happen? I'll give you an example here. Nodi, ivaga DNA ali, let's say, it is a nucleoside. That means we have nitrogenous base. So, here we have DNA, oh sorry, we have sugar. The sugar is going to be deoxyribose sugar. Okay, deoxyribose sugar. Now that sugar plus what, let's say adenine is there, because nitrogenous base is alva. So let's say plus adenine is there. So what will be the name of it? We will call it deoxy. Okay, what will you call it? Deoxy adenosine. Okay, adenosine. So ine idre, that means that is a nucleoside. Okay, it is a nucleoside. Now let's take, students, let's say one more we will take. Let's say thymine is there. Okay, deoxyribose sugar plus thymine is there. Then what do we call it? We call it deoxythymidine. ಆ ಥರ ನಿಮಗೆ ಯೂರಿಡಿನ್, ಸೈಟಿಡಿನ್ ಆ ಥರ ಎಲ್ಲ ಬರಿಬೋದು. ಓಕೆ, ಯೂರಿಡಿನ್, ಸೈಟಿಡಿನ್ ಬರಿಬೋದು. ನಾವು ಲೆಟ್ಸ್ ಸೇ ನಮಗೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇದೆ. ಓಕೆ, ನಮಗೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇದ್ರೆ ವಾಟ್ ವಿಲ್ ಬಿ ದ ಸಫಿಕ್ಸ್ ಮ್ಯಾಮ್? ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇದ್ರೆ ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ 8. Who ate? I ate. Okay, 8. ATE. Okay, so students, nucleotide idre, what is it going to be? It is going to be deoxy sugar plus nitrogen base. Okay, now which nitrogen base you want me to take here? Adenine only we'll take. Okay, adenine plus illi phosphate group is going to be there, right? Phosphate is going to be there. So that will together form deoxyadenylate. Okay, ಇಲ್ಲಿ adenosine ಇತ್ತು, ಇವಾಗ 8 ಇದೆ ಅಲ್ವಾ? So 8 is going to be ಅಂದ್ರೆ adenylate ಅಂತ ಬರುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ ನಿಮಗೆ? Okay, so it is going to be adenylate. See, 8 is going to come in there. So ಈ ತರ ಅವರೊಂದು ಬಾಕ್ಸ್ ಅಲ್ಲಿ ನೀಟಾಗಿ ಕ್ವೆಶ್ಚನ್ ಹೇಳ್ತಾ ಇದ್ದೀನಿ. ನೀಟ್ PYQ ಇದು. ಓಕೆ, ನೀಟ್ PYQ ಇಲ್ಲೇನಾಗುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್? ಸಿ, ನೀಟ್ PYQ ಅಲ್ಲಿ ಅವರು ಥೈಮಿಡಿನ್, ಡಿಆಕ್ಸಿಸೈಟಿಡಿನ್, ಡಿಆಕ್ಸಿಅಡಿನೈಲೈಟ್ ಅಂತ ಮಿಕ್ಸ್ ಮಾಡಿಕೊಡ್ತಾರೆ ಒಂದು ಬಾಕ್ಸ್ ಅಲ್ಲಿ. ಅದಾದ್ಮೇಲೆ ಅವ್ರು ನಿಮಗೆ ಹೇಳ್ತಾರೆ ಪ್ಲೀಸ್ ಐಡೆಂಟಿಫೈ ದ ನಂಬರ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್, ದ ನಂಬರ್ ಆಫ್ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ಸ್. So how will you identify it? You will identify it with ine in the end. Okay, ine is there, 8 is there, like that you will identify it. Okay, these are all neat questions. Okay, so all that detail I'll explain it here, so you don't have to worry. Done? Clear all of you? Is there anyone who has not understood? Now I'm going to teach you little more additional concepts, so that's why I want all of your attention very, very importantly. Okay, very importantly. Now we'll come to this. Before that, I want to teach you one more concept. Okay, one more concept I'll teach you here. Look here, all of you. Now you'll be thinking, ma'am, how does two nucleotides, okay, how does two nucleotides actually combine with each other, right? You'll be thinking this doubt, how does two nucleotides combine with each other? Now listen here carefully, students. We have to draw one whole set for that. Okay, here we have O. Okay, one second, I think it's too thick. Sometimes no, this board, this pen no doesn't do the job very well. Okay, but we will try to make the job done. So first you draw sugar. Okay, and you do the same thing here. First you do the bonding. Okay, first you do the bonding. So illi, let's say we have a purine Okay, we have a purine. Now illi from here to here, okay, from here to here there is going to be one glycosidic bond. Okay, then from here to here, okay, we will have a phosphate. Okay, we'll have a phosphate here, correct? This is done. Now what you're going to do, now how does one bind with another one, right? One will bind with another one. How? Now let's say these two people have to bound together. These two people have to bind together. How will they bind? Have you thought about it? Have you thought about it? Yes or no? So students, they are going to bind. Let's say idon nucleoside, alva. Sorry, idon nucleotide. Idino nucleotide. So one nucleotide has to combine with another nucleotide. Adike correctly young formation agutte ma'am. So nodbek students, illi nodi artha madkoli, illi phosphate ide alva. One nimsha, illi full phosphate draw martini. Okay, so let's say we have I have to draw straight line only. Okay, just give me a minute, I will draw it aram se. Okay, Arjun, if I draw, you will not understand it. Okay, 1 second. We have a sugar. Okay, never mind, I'll not draw that. Okay, now sugar done. Now sugar is going to bind, form a bond, okay, with who? Our purine. Okay, so here, 1 minute. Okay, yes, bonding done. Now illi phosphate alva. So we will just complete the diagram. So here you see students, you have OH, H, 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 OH, H, CH2, O. Okay. Now to this O you have phosphate, double bond O, O and O. Okay. Now this is the structure. Yes or no? Come on students, respond in the chat box. Yakish silent agidira. Respond in the chat box quickly. If you respond only, I will know that you are understanding or not. Okay, yes. So here 2 bonds I have highlighted with red color. So this is one nucleotide. Okay, let's just duplicate this nucleotide. Let's erase this nucleotide. Okay, because il phosphate I have to show illandre niಮಗೆ ಅರ್ಥ ಆಗಲ್ಲ. Okay. Okay, ಇಲ್ಲೊಂದು nucleotide. ಈ nucleotide ಇಲ್ಲಿ ಶಿಫ್ಟ್ ಮಾಡೋಣ. ಓಹೋ, ಎಷ್ಟು ಫಾರ್ಮ್ ಆಗಿದೆ. Okay, now here. Done. This is a nucleotide, this is a nucleotide. Done, done. So, ಇಲ್ಲಿ ನೋಡಿ. Let's say this is nucleotide 1, this is nucleotide 2. Okay, nucleotide 2. Now, what do you notice here? What do you notice here? You have to make a bond. ಇಲ್ಲ ಈ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಮತ್ತೆ ಈ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಬಾಂಡಿಂಗ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕು. ಬಾಂಡ್ ಫಾರ್ಮ್ ಮಾಡಿಲ್ಲ ಅಂದರೆ how do you form the entire nucleic acid? How do you form the entire polynucleotide chain ಅಲ್ವಾ? ಸೊ ಅದಕ್ಕೆ ಅಲ್ಲಿ ಫಾರ್ಮೇಷನ್ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್. ಇವಾಗ ನೀವು ವಿಲ್ ಬಿ ಥಿಂಕಿಂಗ್ ಮ್ಯಾಮ್ ಯಾರ ಜೊತೆ ಫಾರ್ಮೇಷನ್ ಆಗೋದು? ಸೊ ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ಫಾಸ್ಫೇಟ್ ಬಂದ್ಬಿಟ್ಟು ಇಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಡೂ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ವಿತ್ ದ 3OH. It is going to do formation with the 3OH and a bond between 2 nucleotides. Okay, remember a bond between 2 nucleotides. A bond between 2 nucleotides is called as phosphodiester linkage or bond. Phosphodiester. Di andre 2. So, illi phosphoester bond. This is phosphoester bond. Ideno phosphodiester bond. Artha aita ellargo? This is phosphodiester bond. So, this is done and dusted. So, now this is how 2 nucleotides are going to make bond with each other. Okay, so we will have, always remember, we will have, illodi, we will have one sugar molecule. Okay, one sugar molecule will be there. Now, this is one sugar molecule. Okay, I have drawn it on one side. Now, to the fifth carbon, you will have one phosphate that will be attached. Okay, one phosphate will be attached. and to the first carbon, okay, here like this way you can just draw, there will be a nitrogenous base that will be attached. ಈ ತರಾನೇ continuous formation ಆಗುತ್ತೆ students, chain to chain, okay, chain to chain they will form like this, okay. This will go and attach to the third carbon. So here you see, here one bond formation will happen, phosphate to the third carbon, same thing again you repeat, okay, you repeat again here, okay, like this the chain will continue, phosphate to the third carbon. So now you remember here idon chain, okay, this is one chain. ಈ chain ಅಲ್ಲಿ ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಈ end ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, what will be there in this end? In this end you can see there is a free phosphate in the 5' end, correct? This is the 5' end. So on the 5' end there will be a free phosphate. There is always a free phosphate in the 5' end, okay. And here what do you see students? Here there is going to be a 3OH that is free, okay, 3OH is free, right? 3OH is free. Now this becomes the 5' end. So normally num DNA strands, how do they run? They run anti-parallelly. So one second, what happened? Yeah. So this is going to be the 5' end of the DNA. This is going to be the 3' end of DNA. So this is how DNA is going to run. Is this clear? Is this clear? Done? Is this clear to all of you? So the DNA runs from 5' to 3'. Now how did we come to a conclusion which is going to be the 5' and which is going to be the 3' end? Students, ಇಲ್ಲಿ ನೋಡಿ, ಈ 5' end ಅಲ್ಲಿ ಒಂದು phosphate free ಆಗಿರುತ್ತೆ, okay. So 5' end ಒಂದು free phosphate ಇರುತ್ತೆ. ಇಲ್ಲಿ ನೋಡಿದ್ರೆ 3' end ಅಲ್ಲಿ ಒಂದು OH free ಆಗಿರುತ್ತೆ. So that becomes the 3' end. This becomes the 5' end. So 5' end to 3' end, it is going to be what? One chain. ಇವಾಗ I mean one strand, okay. Opposite strand ಹೆಂಗಿರುತ್ತೆ? ಇಲ್ಲಿಂದ ಹಿಂಗೆ ಹೋಗುತ್ತೆ. That is why we say the DNA is parallel, anti-parallel. So ಇಲ್ಲಿ 3' ಇರುತ್ತೆ, ಇಲ್ಲಿ 5' ಇರುತ್ತೆ. So this is how the DNA strands are going to run anti-parallel to each other. I hope this is clear to all of you. So between two nucleotides, okay, between two nucleotides, this is nucleotide 1 and this is nucleotide 2. Phosphodiester bond. Phosphodiester bond is going to be formed, okay. Phosphodiester bond is going to be formed. Done? All of you? Artha aita? Yes, understood? Awesome, awesome. Perfect. Okay, diagram is perfect. Don't worry, ಇನ್ನೂ ಕಲರ್ಸ್ ಇರುತ್ತೆ ನಿಮ್ಮ PDF ಶೇರ್ ಮಾಡೋ ಟೈಮಲ್ಲಿ. ಇವಾಗ ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಕಲರ್ಸ್ ತೋರಿಸ್ತಾ ಇದೆ. Very good. Okay, let's mark more attendance. Okay, ಬೇಗ ವಿಡಿಯೋಗೆ ಒಂದು ಲೈಕ್ ಕೊಡಿ. Smash the like button. Okay, we have some messages. Okay, awesome. Devi has put a lot of hearts. Yes, very. Oh. My God, the hearts are not so many hearts. So many hearts that cannot be seen. Yes, clear? Clear, dear? Awesome. Okay, done. Sahana, yes. Okay, perfect. Okay, perfect. Students, ನೀವು ಮಾತಾಡಿಲ್ಲ ಅಂದರೆ ನನಗೆ ತುಂಬ ಕಷ್ಟ ಆಗುತ್ತೆ. Okay, I will not know ನಿಮಗೆ ಅರ್ಥ ಆಗ್ತಾ ಇದೆಯಾ ಇಲ್ವಾ, ರಿಪೀಟ್ ಮಾಡಬೇಕಾ? Okay, ಆ ಥರ ಫಾರ್ಮ್ಯಾಲಿಟೀಸ್ ನನ್ನ ಕ್ಲಾಸಲ್ಲಿ ಇರಬಾರ್ದು. ಆಲ್ ಆಫ್ ಯು ಶುಡ್ ಬಿ ಫ್ರೀ ಟು ಟೆಲ್ ಮಿ ಎಸ್ ಮ್ಯಾಮ್ ಐ ಡಿಡ್ ನಾಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮ್ಯಾಮ್ ಪ್ಲೀಸ್ ರಿಪೀಟ್ ಮಾಡಿ ಮ್ಯಾಮ್. ನಾನು ಇದು ಮಾಡೋದು ನಿಮಗೆ. ಸೊ ಇಫ್ ಯು ಡೋಂಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಆರ್ ಇಫ್ ಯು ಹೆಸಿಟೇಟ್ ಟು ಆಸ್ಕ್ ಮೀ ಟು ರಿಪೀಟ್ ದೆನ್ ವಾಟ್ ಇಸ್ ದ ಯೂಸ್ ಆಫ್ ದ ಕ್ಲಾಸ್ ಅಲ್ವಾ? ಸೊ ಇಲ್ಲಿ ನೋಡಿ ನಮಗೆ NCERT ಲೈನ್ಸ್ ನೋಡೋಣ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ in RNA every nucleotide residue has an additional OH group present. Now students, NEET ಅಲ್ಲಿ biology crack ಮಾಡಕ್ಕೆ ಸ್ವಲ್ಪ ಕಷ್ಟ ಅಂತ most of you will know it that biology ಮ್ಯಾಮ್ ಸ್ವಲ್ಪ ಕಷ್ಟ ಇದೆ. ಓಕೆ, ಸ್ವಲ್ಪ ಒಂದು tiny bit is little difficult, ಒಂದು ಕುಟ್ಟಿ bit is little difficult. Why is that happening? Because ನೋಡಿ ಈ ಪ್ಯಾರಾಗ್ರಾಫ್ ನಾನು ಫಸ್ಟ್ ಡೇ ಲೆಟ್ಸ್ ಡೈರೆಕ್ಟ್ಲಿ ನಾನು NCERT ಓಪನ್ ಮಾಡಿ ಓಕೆ ಇವತ್ತು ನಾನು ಓದಬೇಕು, ನೆಕ್ಸ್ಟ್ ಲೆವೆಲ್ ನಾನು ಓದ್ತೀನಿ ಅಂತ ಸ್ಟಾರ್ಟಿಂಗಿಂದಾನೇ ನೀವು NCERT ಓದಕ್ಕೆ ಶುರು ಮಾಡಿದ್ರೆ ನಿಮಗೇನು ಅರ್ಥ ಆಗಲ್ಲ. ಫಸ್ಟ್ ನೀವು ಯಾವಾಗಲೂ ನೀಟ್ ಪ್ರಿಪ್ರೇಷನ್ಗೆ ಫಸ್ಟು ಲೆಕ್ಚರ್ ನೋಡಬೇಕು. ಒಂದು ಚೆನ್ನಾಗಿರುವ ಟೀಚರ್ದು ಲೆಕ್ಚರ್ ನೋಡಿಬಿಟ್ಟು ವಾಪಸ್ NCERT ಓದಿ ವಾಪಸ್ ಇದು PYQ ಸಾಲ್ವ್ ಮಾಡಿದ್ರೆ ಒಂದು ಪ್ರಾಪರ್ ಸಿಸ್ಟಮ್ ಇರುತ್ತೆ. ಬಿಕಾಸ್ ಫಸ್ಟ್ ನೀವು ಎಲ್ಲ ಕಾನ್ಸೆಪ್ಟ್ಸು ಕ್ಲಿಯರ್ ಆಗಿ ಒಂದು ಫೌಂಡೇಶನ್ ಇಂದ ಯು ಹ್ಯಾವ್ ಅಂಡರ್ಸ್ಟುಡ್. ಅದಾದಮೇಲೆ ಯು ವಿಲ್ ರೀಡ್ NCERT, ಅದಾದಮೇಲೆ ಕ್ವಶನ್ ಪ್ರಾಕ್ಟೀಸ್. ಸೊ ಈ ಥರ ಒಂದು ಫೌಂಡೇಶನ್ ಬಿಲ್ಡ್ ಮಾಡಬೇಕು. ಫಸ್ಟೇ NCERT ಓದಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರೆ ಇಟ್ ವಿಲ್ ಬಿ ಅ ಬಿಲ್ಡಿಂಗ್ ವಿತೌಟ್ ಲೈಕ್ ಸಿಮೆಂಟ್ ಆ್ಯಂಡ್ ಬ್ರಿಕ್ಸ್. ಸೊ ಆ ಥರ ಮಾಡಬೇಡಿ. ಕರೆಕ್ಟಾಗಿ ಏನು ಮಾಡಬೇಕು, ಫಸ್ಟ್ ಒಂದು ಲೆಕ್ಚರ್ ನೋಡಿ, ಚೆನ್ನಾಗಿರೋ ಒಂದು ಲೆಕ್ಚರ್ ನೋಡಿ, ನಿಮಗೆ ಅರ್ಥ ಆಗೋ ಥರ ಒಂದು ಟೀಚರ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತಾ ಇದ್ರೆ ಆ ಒಂದು ಲೆಕ್ಚರ್ ನೋಡಿ, ಅದಾದಮೇಲೆ NCERT ಓದಿ, ಅದಾದಮೇಲೆ PYQs ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡಿದ್ರಿ. ಈ ಥರ ಒಂದು ಸೆಟ್ಟಲ್ ಮಾಡಿದ್ರೆ ನಿಮಗೆ ಬಯಾಲಜಿಯಲ್ಲಿ 360 ಖಂಡಿತ. ಓಕೆ, ಸೊ ಲುಕ್ ಹಿಯರ್ ವಿ ಹ್ಯಾವ್ in RNA every nucleotide residue has an additional OH group present at the second position, okay, in the ribose. Also in RNA the uracil is found at the place of thymine. So students, thymine ge 5-methyl uracil anta helbodu. This is an AIPMT level question. AIPMT question. Okay, little advanced question. Okay, now another chemical name of thymine, 5-methyl uracil. Andre uracil alli 5 position alli methyl add madre, that becomes thymine. DNA as an acidic substance. Okay, DNA is acidic. Why? Because of the phosphate. DNA is an acidic substance present in the nucleus, and it was first identified by Friedrich Mischer in 1869. In what year? 1869. He named it as nuclein. He named it what? Nuclein. However, due to the technical limitations in isolating such a long polymer intact, the elucidation of the structure of DNA remained elusive for a very long time. Andre ಅದನ್ನ ಒಂದು ಅದರದ್ದು ಸ್ಟ್ರಕ್ಚರ್ ಒಂದು ಐಡೆಂಟಿಫೈ ಮಾಡಕ್ಕೆ ಸ್ವಲ್ಪ ಟೈಮ್ ಎತ್ಕೊಂಡಿದೆ. ಓಕೆ, ದಟ್ ಈಸ್ ಇಟ್ ಟುಕ್ ಅ ಲಾಟ್ ಆಫ್ ಟೈಮ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ DNA. ವೈ? ಬಿಕಾಸ್ ಇಟ್ ವಾಸ್ ಟೂ ಲಾಂಗ್ and there were some technical limitations. Let's say I want to do a 12 hours marathon on YouTube. There's a technical limitation on YouTube that you cannot take a 12 hours marathon. Okay, you can only take 10 hours marathon. Illandre on 10 hours live ಹೋಗಬಹುದು, ಇಲ್ಲಾಂದ್ರೆ 11 hours ಹೋಗಬಹುದು ಅಂತ. ಬಟ್ ಲೆಟ್ಸ್ ಸೇ 12 hours ಆಗುತ್ತೆ, ಬಟ್ 18 hours ಆಗಲ್ಲ. ಸೊ ಆ ಥರ ಒಂದು ಟೆಕ್ನಿಕಲ್ ಲಿಮಿಟೇಷನ್ ಇತ್ತು ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ DNA ಸ್ಟ್ರಕ್ಚರ್. ಓಕೆ, ಹಾಯ್ ನಂದಿನಿ, ಹೌ ಆರ್ ಯು? ಸೊ however, due to the technical limitation in isolating such a long polymer intact, the structure, like adon finding ಸ್ವಲ್ಪ it took time. Okay, now it was only in 1953 that James Watson and Francis Crick, based on X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but a famous double helical model for the structure of DNA. One of the hallmark of their proposition was base pairing between the two strands. Now I'll explain this base pairing here. Okay, now students, two DNA strands are there, no? So illon DNA strand draw madana. Okay, this is one. This is one DNA strand. Okay, this is one DNA strand, and we have complementary strand. I'll draw it in a little different color. This is the DNA strand, correct? Yes ma'am, these are the two strands. Okay, these are the two strands. Yes, happy evening. Okay, now these two strands are there. Now look here, here you say adenine is there. Okay, here let's say thymine is there, here cytosine is there, here guanine is there, here let's say guanine, cytosine, adenine, thymine, adenine, thymine. Like this, something is there on the strand. Now, opposite strand in madbeko, complementary base pair madbeko. Complementary base pair it has to make. That means, remember this trick. The trick is, students, at Gopika Ma'am's class. Okay, Guru Bhakti should be in your head, so you can remember Ma'am in your head. But illa andre at Gopika Ma'am's class. A, T, G and C. That means adenine always binds with thymine. At, okay, so this together is going to become at. This is going to be Gopika's, okay, class. Okay, Gopika Ma'am's class. Got it? So remember, adenine always binds with thymine and guanine always binds with cytosine. Keep this in your mind and you tell me the other answers. Illi ಏನು ಬರುತ್ತೆ students? ಇಲ್ಲಿ ನೋಡಿ, adenine will always bind with whom? Thymine. Thymine. Thymine will always bind with adenine. Cytosine will always bind with guanine. Guanine will always bind with cytosine. Cytosine, guanine, thymine, adenine, adenine, thymine, thymine, adenine. Correct alva? Check ಮಾಡಿ ಹೇಳಿ. At Gopika Ma'am's class ಬರಬೇಕು. Okay, at ಬಂದಿದೆ. TA ಬಂದಿದೆ. Gopika Ma'am's class ಬಂದಿದೆ. Gopika Ma'am's class, Gopika Ma'am's class. Okay, C and G. Okay, Gopika Ma'am's class. At has come, TA has come, at has come, TA has come. So this is called as complementary base pairing. That means one strand, let's say, is running from 5 dash to 3 dash. One more strand is running from 3 dash to 5 dash. Okay. Now this is called as base pairing. Andre nitrogenous bases main job ಏನಂದ್ರೆ ಅವರು ಈ ಎರಡು DNA strands ನ ಒಟ್ಟಿಗೆ ಇರಬೇಕು. Okay, they have to keep it together. Okay, they have to keep it together. So I'm erasing this. Okay, so first salient feature. Okay, students, first salient feature of DNA is that DNA structure. Okay, DNA's backbone. Okay, DNA structure's backbone is made up of, okay, is made up of what? It is made up of sugar and phosphate. Made up of what? Sugar and phosphate. Now you'll be thinking, huh? Where is nitrogenous base? And look here, bases, okay, bases project. ನಿಮ್ಮ NCERT lines ಇದೆಲ್ಲ projects inwards. Projects inwards. That means what? Let's assume this is one DNA strand, this is one DNA strand. Okay, watch ಹಾಕಿಲ್ಲ students, but ring ಅಲ್ಲಿ identify ಮಾಡಿ. Okay, ಇದು remove ಮಾಡ್ತೀನಿ. Okay, ಇವಾಗ ಎರಡು ಕೈಯಲ್ಲೂ ring ಇದೆ ನೋಡಿ, ring. Okay. Okay, now this is one hand. Okay, this is one DNA strand. Okay, this is running from 5 dash to 3 dash. This is from 3 dash to 5 dash. ಈ ಎರಡು DNA strands, so backbone ಇಲ್ವಾ? This is the backbone. ಇವಾಗ ನನ್ನ ಕೈಯಲ್ಲಿ bones ಇದೆ ಅಲ್ವಾ? ನಿಮ್ಮ ಕೈಯಲ್ಲೂ bones ಇದೆ. But ಇಲ್ಲಿ, let's say this is the backbone. 5 dash to 3 dash, 3 dash to 5 dash ಅಲ್ವಾ? This is backbone is made up of what? ನಾನು ಹೇಳ್ತಾ ಇದ್ದೀನಿ, oh your ma'am, ನಿಮ್ಮ ಹ್ಯಾಂಡು humerus, radius ಅಲ್ನ carpels, metacarpels ಇಂದ ಫಾರ್ಮ್ ಆಗ್ತಿದೆ. ಅದರನೇ DNA is formed of what? Sugar ಮತ್ತೆ phosphate. ಇಲ್ಲೂ ಏನು students? Sugar and phosphate. So this is the backbone of the DNA. Now to connect this both, now to connect my this hand and this hand, I need someone who is projecting inwards. So that is going to be your nitrogenous bases. So adenine will bind with thymine like this. They are going to connect with each other and they are going to form that X-ray. That is the double helical structure. Clear? Thumbs up please in the chat. Okay. So this is done. DNA's backbone is made up of sugar and phosphate and bases project inwards. Now next salient feature of DNA is what? DNA is a double helical structure. DNA has a double. DNA has a double helix structure. Okay, double helical structure. Got it? Double helical structure. Now this double helical structure, okay, this double helical structure discovery ಹೆಂಗಾಗಿದೆ? How did it happen ಅಂತ ಇಲ್ಲಿ explain ಮಾಡಬೇಕು. So here remember, double helical structure was given by Watson 4 people, okay? Crick, Roslyn, Franklin, and Wilkins. Okay? These 4 people, students, these 4 people gave the DNA double helical structure. So, these 4 people were like, okay, what did these 4 people do? They did something called as X-ray diffraction. They did something called as X-ray diffraction. They did something called as X-ray diffraction. So, in X-ray diffraction, they understood that nam DNA bandbit on double helical structure anta aurgattaitu. Okay? DNA bandbit ಏನು students on double helical structure anta aurgattaitu. Hengattaitu with the help of your this one. Nodi ಇಲ್ಲಿ X-ray diffraction ಇಂದ ಅವ್ರಿಗೆ ಅರ್ಥ ಆಯಿತು DNA ಈ ತರ double helical structure ಆಗಿರುತ್ತೆ ಅಂತ. Understood? Yes or no? Yes or no? Understood, is it? All of you? Yes, ma'am. Awesome. Okay. Double helical structure, you have to remember who gave all the double helical structure, okay? Because here we are going to study further about the double helical structure and other things. So, this ಸ್ವಲ್ಪ ಈ side draw ಮಾಡಬೇಕಾಗಿತ್ತು, but it's okay. Now, students, this type of DNA, okay, this type of DNA is called B-type DNA. Okay, this DNA that we study, no, is called as B-type DNA. Either A-type DNA there, Z-type DNA there. So, we are studying the B-type DNA form. B-type DNA form, and this DNA is also in the right-handed fashion. Okay, this DNA is also in the right-handed fashion. Right-handed fashion. Done? Okay, awesome. Now we will write the next point. Okay, we will write the next point for your double, that is your salient features of DNA. Salient features of DNA. Okay. Now here, okay, now here you write that the DNA structure is anti-parallel. It has anti-parallel polarity. What it has? It has anti-parallel polarity. Andre students, ill nodi, what is the meaning of it? One is running from 5' to 3', one more is running from 3' to 5'. Okay, that is called as anti-parallel polarity. Done? Very good. Okay. Now, about base pairing also you have to write. So, what you will write here, base pairing? Remember, base pairing, okay, base pairing, you will remember that adenine always, okay, always binds with thymine, okay, thymine with 2 hydrogen bonds and guanine, Gopika Mam's class, okay, guanine binds with whom? Class alva, with cytosine. Cytosine binds with cytosine with 3 hydrogen bonds. Arthaita? 3 hydrogen bonds. Okay? Good job. Okay. Ivaga salpa numbers ide students. So, 1 turn of DNA. Okay, remember? 1 turn of DNA has around 10 base pairs. Okay, 1 turn of DNA has around 10 base pairs. Andre, itara on DNA, okay, 1 turn alli 10 base pairs irutte. ಲೈಕ್ 1 2 3 4 5 6 7 ಆ ಥರ 10 ಬೇಸ್ ಪೇರ್ಸ್ ಇರುತ್ತೆ. ಅಂಡರ್ಸ್ಟುಡ್? 10 ಬೇಸ್ ಪೇರ್ಸ್ ಇನ್ ದಿಸ್ ಒನ್. ನಾವು ಡಿಸ್ಟೆನ್ಸ್ ಬಿಟ್ವೀನ್, ಸೊ ಲೆಟ್ಸ್ ಸೇ ಈ ಬೇಸ್ ಪೇರ್ ಇಂದ ಈ ಬೇಸ್ ಪೇರ್ಗೆ ಎಷ್ಟು ಡಿಸ್ಟೆನ್ಸ್ ಇದೆ ಅಂತ ಗೊತ್ತಿರಬೇಕು ನಮಗೆ. ಸೊ ದ ಡಿಸ್ಟೆನ್ಸ್ between base pair is equal to how much, students? Distance between base pair, anyone in the chats? Distance between two base pairs is going to be 0.34 nanometer. 0.34 nanometer. ಇದರಿಂದ ನಮಗೆ ಹೆಲಿಕ್ಸ್ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡ್ಬೋದು, ಇಲ್ಲಾಂದ್ರೆ ಪಿಚ್ ಆಫ್ ದ DNA ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡ್ಬೋದು. ಜಸ್ಟ್ ಮಲ್ಟಿಪ್ಲೈ ದಿಸ್ ಬೋತ್. ಸೊ ಪಿಚ್ ಆಫ್ DNA, okay, is equal to 3.4 nanometer. 3.4 nanometer. So these numbers are important. These numbers are important. Done, done, done. Done, done, done. Okay. ಇವಾಗ ಸ್ವಲ್ಪ PYQ solve ಮಾಡಿಸ್ತೀನಿ. ಅದಕ್ಕೆ ಮುಂಚೆ I'll teach you Chargaff rule and then I'll make you solve PYQs. Okay. Okay. First I'll do Chargaff rule because the first question is based on the Chargaff rule. Yes, 0.34 nanometer. Excellent, excellent. Okay, good job. Okay. Next time in the use ಸ್ವಲ್ಪ dark background ಆಗ್ತೀನಿ so that the reflection will be less. Okay, reflection will be less. Okay, awesome. Now let's continue. Okay, let's continue. So students, Chargaff rule. Now what do we know about Chargaff? So Chargaff was a person. So his full name is Erwin Chargaff. Erwin Chargaff. You. Okay, Erwin Chargaff. So his rule was that, so let's imagine, okay, ನೀವೇ ಹೇಳಿ, okay, ನಿಮ್ಮ ಕ್ಲಾಸಲ್ಲಿ, okay, 4 ಬಾಯ್ಸ್ ಇದ್ದಾರೆ, okay, 4 ಬಾಯ್ಸ್ ಇದ್ದಾರೆ, okay, 4 ಬಾಯ್ಸ್ ಇದ್ದಾರೆ, 4 ಗರ್ಲ್ಸ್ ಇದ್ದಾರೆ. ಇವಾಗ ಸ್ಟೂಡೆಂಟ್ಸ್ ಲೆಟ್ಸ್ ಸೇ ನನಗೊಂದು ಆ್ಯನ್ಯುವಲ್ ಡೇ ಡ್ಯಾನ್ಸ್ ಮಾಡಬೇಕು, ಇಲ್ಲಾಂದರೆ ಏನಾದರೂ ಒಂದು ಸ್ಪೆಷಲ್ ಪ್ರೋಗ್ರಾಮ್ ಇದೆ, ನನಗೆ ಪೇರ್ಸ್ ಮಾಡಬೇಕು, ಒಂದು ಬಾಯ್ ಒಂದು ಗರ್ಲ್, ಒಂದು ಬಾಯ್ ಒಂದು ಗರ್ಲ್ ಅಂತ. ಆಬ್ವಿಯಸ್ಲಿ ಇಲ್ಲಿ ಕ್ಯಾಲ್ಕುಲೇಷನ್ ತುಂಬ ಈಸಿ ಆಗುತ್ತೆ. ವೈ? ಬಿಕಾಸ್ ದಿಸ್ ಬಾಯ್ ವಿಲ್ ಬಿ ದಿಸ್ ಗರ್ಲ್ಸ್ ಪಾರ್ಟ್ನರ್, ದಿಸ್ ಬಾಯ್ ವಿಲ್ ಬಿ ದಿಸ್ ಗರ್ಲ್ಸ್ ಪಾರ್ಟ್ನರ್, ದಿಸ್ ಬಾಯ್ ವಿಲ್ ಬಿ ದಿಸ್ ಗರ್ಲ್ಸ್ ಪಾರ್ಟ್ನರ್, ದಿಸ್ ಬಾಯ್ ವಿಲ್ ಬಿ ದಿಸ್ ಗರ್ಲ್ಸ್ ಪಾರ್ಟ್ನರ್. ಕರೆಕ್ಟ್ ಅಲ್ವಾ? ಸೊ ಎಷ್ಟು ನಂಬರ್ ಆಫ್ ಬಾಯ್ಸ್ ಇತ್ತು ಅಷ್ಟು ನಂಬರ್ ಆಫ್ ಗರ್ಲ್ಸು ಇತ್ತು. ಸೊ ಇಲ್ಲಿ ರೇಷಿಯೋ ಪ್ರಾಬ್ಲಮ್ ಬಂದಿಲ್ಲ, ರೇಷಿಯೋಸ್ ವರ್ ಪರ್ಫೆಕ್ಟ್. But let's say same scenario dalli, let's say we have 5 or let's say 6 girls and we have only 4 boys. 2 girls will be left alone or pair iralla. So that means ratio goes wrong. So now Chargaff was like, wait, if you are saying that the DNA structure, ilva, both the strands are complementary to each other and the base pairing is very necessary, then the number of purines should be equal to the number of pyrimidines. We were like, yes, Eureka, correct agide alva. So he told that the ratio, now here I want all of your attention, ratio of purine means what am I talking about? Adenine and guanine. Okay, ratio of purines will always be equal to the ratio of pyrimidines. Ratio of pyrimidines. Okay, ratio of pyrimidines andre here what am I talking about? Cytosine, uracil matte thymine. Okay, so that means I'm saying that how many purines are there, that many pyrimidines should be there, so that adenine will always bind with thymine with 2 hydrogen bonds and guanine will always bind with cytosine with 3 hydrogen bonds. So this is very important alva. Now you will be asking me, ma'am, yak ma'am adenine guanine bind ಮಾಡಲ್ಲ, cytosine uracil bind ಮಾಡಲ್ಲ? Students, there is a simple thing alva. ಇವಾಗ ಪ್ಯೂರಿನ್ ಸ್ಟ್ರಕ್ಚರ್ ಬಂದ್ಬಿಟ್ಟು 9 ಮೆಂಬರ್ಡ್ ರಿಂಗ್, ಒಂದು ದೊಡ್ಡ ಸ್ಟ್ರಕ್ಚರ್. ನಾವು ದ್ಯಾಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಸಿಂಕ್ ವಿತ್ 6 ಮೆಂಬರ್ಡ್ ರಿಂಗ್ ಅಲ್ವಾ? ಆ ಥರನೇ ಇಲ್ಲೂ ಆ ಥರನೇ 9 to 6, 6 to 9. ಸೊ ಅಂದರೆ ಆಲ್ಮೋಸ್ಟ್ ನೋಡಿದ್ರೆ ಅವ್ರ ಶೇಪ್, ಡಿಫ್ರೆನ್ಸ್ ಬಿಟ್ವೀನ್ ಬೋತ್ ದ DNA, ಡಿಸ್ಟೆನ್ಸ್ ಬಿಟ್ವೀನ್ ಬೋತ್ ದ DNA ಸ್ಟ್ರಾಂಡ್ಸ್ ಎಲ್ಲ ಒಂಥರ ಸಿಮಿಲರ್ ಆಗಿರುತ್ತೆ. ಬಟ್ ಲೆಟ್ಸ್ ಸೇ ನಾನು ಪ್ಯೂರಿನ್ 9 to 9, 9 to 9, 6 to 6 ಮಾಡಿದ್ರೆ, ಲೆಟ್ಸ್ ಜಸ್ಟ್ ಇಮ್ಯಾಜಿನ್. ಓಕೆ, ಇವಾಗ DNA structure. Okay, idella ಸ್ವಲ್ಪ detail ಆಗಿ explain ಮಾಡ್ತಾ ಇದೀನಿ. So you can answer assertion reason question for NEET. Okay, ಈ structure ನೋಡಿ students equal ಆಗಿರುತ್ತೆ, binding problem ಇರಲ್ಲ. ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಇಲ್ಲೂ ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಇಲ್ಲಿ ಚಿಕ್ಕದಾಗಿರುತ್ತೆ, ಇಲ್ಲೂ ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಚಿಕ್ಕದಾಗಿರುತ್ತೆ ಸ್ಟ್ರಕ್ಚರ್ ಅಲ್ವಾ? ಬಿಕಾಸ್ 9 ಅಂದರೆ ಬಾಂಡಿಂಗ್ ತುಂಬ. ಓಕೆ, ಲೆಟ್ಸ್ ಸೇ ಇಲ್ಲಿ ಎರಡು ಸೇಮ್ ಆಗಿದೆ, ಇಲ್ಲಿ ಬಂದ್ಬಿಟ್ಟು ಈ ಥರ ಇರುತ್ತೆ. ಓಕೆ, ಲೆಟ್ಸ್ ಸೇ ಇನ್ನೂ ಸ್ಟ್ರೈಟ್ ಆಗಿರುತ್ತೆ. ಡು ಯು ಫೀಲ್ ದ ಸೈಜ್ ವಿಲ್ ಬಿ ಕರೆಕ್ಟ್? ಡು ಯು ಫೀಲ್ ದ ಡಿಸ್ಟೆನ್ಸ್ ವಿಲ್ ಬಿ ಕರೆಕ್ಟ್? ಡು ಯು ಥಿಂಕ್ ದ ಡಿಸ್ಟೆನ್ಸ್ ಬಿಟ್ವೀನ್ ದ ಬೇಸ್ ಪೇರ್ ವಿಲ್ ಬಿ ಕರೆಕ್ಟ್? ಇಲ್ಲ, ಕಂಪ್ಲೀಟ್ಲಿ ಚೇಂಜ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ವೈ ಆಲ್ವೇಸ್ ಅ ಪ್ಯೂರಿನ್ ವಿಲ್ ಬೈಂಡ್ ವಿತ್ ಅ ಪಿರಮಿಡಿನ್ and pyrimidine will bind with a purine, and it's not purine and pyrimidine. This is clear to all of you? Little detailed information for all of you. So now students, this we understood, ma'am. Now to explain this, you will be asked questions, okay, for NEET. So NEET questions will be in such a way, okay, I want all of you to answer. The question will go something like this. Let's say cytosine, okay, is equal to 20%. Okay, calculate adenine. ಈ ಥರ ಕ್ವೆಶ್ಚನ್ ಬಂದರೆ ನೀವು ಹೆಂಗೆ ಆನ್ಸರ್ ಮಾಡ್ತೀರಾ? ಬಿಕಾಸ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಇಲ್ಲಿ ನೋಡಿ, ಆಲ್ವೇಸ್ ರಿಮೆಂಬರ್ A2, okay, the number of purines, okay, A and G will always be equal to CUT. Okay, CUT. That means A to T and G to C will be equal to 1, or in other words I can write, this I already wrote, no? A to T will be equal to 1, okay? G to C will be equal to 1. Correct? Because they both have to be the same ratio. That means purine to pyrimidine has to be equal. Now can you calculate this, anyone? So cytosine 20%, calculate adenine anta kottidare. So idana calculate madana. So cytosine 20. So bonding illa again at Gopika Mam's class. What we have cytosine is equal to 20%. So let's say cytosine 20% idre, obviously guanine 20% irutalva. So I am writing guanine also to be 20%. Okay, so let's say this is given to us. So if cytosine is 20, I know guanine is 20. These both are contributing to 40%. Rest how much I have? 60% balance irodu. So ಆ 60% ಅಲ್ಲಿ adenine ಬಂದ್ಬಿಟ್ಟು 30% ಇರುತ್ತೆ. adenine 30% ಇದ್ರೆ automatically time ಇನ್ನು 30% ಆಗಿರುತ್ತೆ. So this will totally give you me 100% ಅಲ್ವಾ, or is equal to 1. So this is how you will calculate your values. So this will give you your Chargaff rule. Now many students ಗೆ ಈ ಒಂದು ಕನ್ಫ್ಯೂಷನ್ ಇರುತ್ತೆ, ಮ್ಯಾಮ್ 80 rich region should always be equal, or let's say 80 rich region is equal to G and C region not equal to. There can be places where this fellow is more, okay, like your micro satellite and mini satellite rich agirbodu. There can be places where this fellow is rich. This fellow is rich. See, it's like, you know, idu couples. Adenine ಮತ್ತೆ thymine ಬಂತರ ಒಂಥರ ಕಪಲ್ಸ್ ತರ, ಅವ್ರು ಕರೆಕ್ಟಾಗಿ ಬಾಂಡಿಂಗ್ ಇರಬೇಕು. ಅವರು ಬೇರೆ ಕಪಲ್ಸ್ ತರ ಸೇಮ್ ರಿಚ್ ಅಲ್ಲಿ ಇರಬೇಕು, ಸೇಮ್ ಪ್ರಾಪರ್ಟಿ ಲೆವೆಲ್ಸ್ ಇರಬೇಕು. ಅವ್ರಿಗೆ 3 ಕಾರ್ ಇದ್ರೆ ನಮಗೂ 3 ಕಾರ್ ಇರಬೇಕು, ಆ ಥರ ಏನೂ ಇಲ್ಲ. ಬಟ್ ಬಾಂಡಿಂಗ್ ಕರೆಕ್ಟ್ ಆಗಿರಬೇಕು. ಅಡಿನೈನ್ ಮತ್ತೆ ಥೈಮಿನ್ ಯಾವಾಗಲೂ ಈಕ್ವಲ್ ಆಗಿರಬೇಕು, ಬಾಂಡಿಂಗ್ ಕರೆಕ್ಟ್ ಆಗಿರಬೇಕು. ಬಟ್ ಅಡಿನೈನ್ ಥೈಮಿನ್ ಗ್ವಾನಿನ್ ಸೈಟೋಸಿನ್ ಈಕ್ವಲ್ ಆಗಿದ್ರೇನು ಇದ್ದಿಲ್ಲ ಅಂದ್ರೆ ನಮಗೆ ಪ್ರಾಬ್ಲಮ್ ಇಲ್ಲ. ಸೊ ದೇರ್ ಕ್ಯಾನ್ ಬಿ ರೀಜನ್ಸ್ ಇನ್ ದ ಕ್ರೋಮೋಸೋಮ್ ಆರ್ ಇನ್ ದ DNA ವೇರ್ AT is going to be more, and there can be regions where GC is going to be more. Understood? That is what you have to understand. That there can be regions where this fellow is more, there can be regions where this fellow is more. But what is important here? Purine and pyrimidine should be in equal ratio. That means how many adenines are there in one strand, that many thymine should be there in this strand, so they both can bond with each other. Is this clear? So this is your Chargaff rule. Okay, so here they have explained. Let's go back here. One of the hallmarks of the preposition was base pairing between the two strands of the polynucleotide chain. However, this preposition was also based on the observation of Erwin Chargaff. Tumba thikka aithe idu. Okay, Erwin Chargaff that for a double stranded DNA, okay, for a double stranded DNA, the ratio between adenine and thymine, guanine and cytosine are constant and equals to 1. What did they say? Ratio between A and T and G and C are constant and equal to 1. Clear? Awesome. Understood? Yes. Okay. Okay. Now we'll solve some PYQs. Okay. We'll solve some NEET. Some are PYQs, some are like little difficult questions. Okay. First question. Which of the following is not consistent with base pairing in double-stranded DNA molecule? The number of C equals the number of G. That is, C equals number of G. Okay. Which of the following is not constant? Okay. Yarella constant agirbardu. The number of A equals T. A equals T. The amount of purine equal to pyrimidine. Purine ratio equal to pyrimidine ratio. Try ಮಾಡಿ answer ಮಾಡೋದಕ್ಕೆ. The ratio of G plus C to A plus G plus C equal to A plus T is same for all species or same for each species. Which one do you think is the answer? Yes, quickly mark your attendance, okay? Quickly mark your attendance and tell me the answer. And students, this video please nim friends jothe share ಮಾಡಿ, whoever is preparing for your NEET. It will help them a lot. Okay? It will help them a lot. Yes or no? See, after I tell only some people will like the video. ಎಷ್ಟು ಕಂಜೂಸ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ನೀವೆಲ್ಲ. Too ಕಂಜೂಸ್. Too ಕಂಜೂಸ್ you people are. 11th classes. Okay, I'll definitely take, okay? Just tell me which chapters you want. Which chapters you want. Option 4. Excellent. Excellent. Okay? Correct. The answer is going to be option number 4 because already explained madidini. Ratio of G plus C and A plus T will not be same for all species. There can be regions where A and T is more. There can be regions where G and C is more. Okay? Good job, students. So, plus 4 marks all of you have got. Who is that? Ashwati, Sahana, okay? Luna, I need animal king. Okay. Rajesh P. Yes, come on. Next question on the screen. Which of the following is not a feature of double helical model of DNA? The two chains have anti-parallel polarity. A purine always comes opposite to a pyrimidine. The pitch of the DNA is 3.4 nanometer. The two chains are coiled in a left-handed fashion. Idu bega answer madi. The two chains are anti-parallel andre. One is running from 5' to 3'. One more is running 3' to 5'. Okay. A purine always comes opposite to a pyrimidine. Yes, a purine will always come opposite to a pyrimidine. Then the pitch of DNA is 3.4 nanometer. The two chains are in a left-handed fashion. Okay. Come on, come on, come on, come on, come on. Come on. Best books. Already nan video madidini dear. Go check KCET 1 year strategy video by Gopika Ma'am. ಆ ವಿಡಿಯೋದಲ್ಲಿ ಎಲ್ಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ, ಬುಕ್ಸ್ ಬಗ್ಗೆ ಎಲ್ಲ ಬಗ್ಗೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ. Yes. Option 4. Very good. Very good. Option 4 is the answer because explain madidine. The two chains are in a right-handed fashion. Right-handed fashion. So plus 8 marks. Okay. Nodi, ill explain madidine. See here, both the chains are in a right-handed fashion. Ill en kelidare, left-handed fashion anta kelidare. So that stays wrong. So you get plus 8 marks. Okay. Plus 8 marks. Next question. The two polynucleotide chains in DNA are parallel, discontinuous, antiparallel, semi-conservative. The two polynucleotide chains in DNA are parallel, discontinuous, antiparallel, semi-conservative. Come on. Come on, come on, come on. Whatever you have studied, only you tell. Okay? Yes, it's okay to make mistakes, students. First class, ido. Make mistakes, learn from your mistake. No one is going to judge you. No one is going to scold you. Okay? Yes, it is just to make you a better aspirant tomorrow. Okay, you're welcome. A video check madbid. I'll try check madi the best books for KCET. Okay? Done? Yes, the answer is option number 3, anti-parallel. Okay, just now only we studied, no? If one is running 5' to 3', one more will run 3' to 5'. Okay, anti-parallel. Parallel level somewhere. Okay, so it is going to be option number 3. Very good. Plus 12 marks. Okay, plus 12 marks. Okay, Ashwati and Sahana has become very, very smart. Okay, yes, good job. Good job. Good, good. Okay, next question. In the DNA molecule, all these are NEET questions, okay? In the DNA molecule, the total amount of purine nucleotide and pyrimidine nucleotide is not always equal. There are two strands which run parallel in 5' to 3' direction. The proportion of adenine in relation to thymine varies with organism. There are two strands which run anti-parallel in 5' to 3' direction and other 3' to 5' direction. Question ಸ್ವಲ್ಪ long ಆಗಿದೆ, okay? But I think we can break down the question. In a DNA molecule, the total amount of purine nucleotides and pyrimidine nucleotides is not always equal against the chaga fruit. There are two strands which run parallel in 5' direction anta kottidare. The proportion of adenine in relation to the proportion of thymine varies anta kottidare. That means at Gopika Mam's class will vary, they are saying. So, what will be the answer? The correct answer will be option number 4. Why? Because there are two strands which run anti-parallelly 5' to 3', 3' to 5'. So, answer is going to be option number 4. That gives you plus 16 marks. Okay. So, next class, please remember this marks. Next class, you please remember this marks and come. Okay. Done and not done. Next question. One turn of helix in a B-form DNA is approximately one turn of a helix in a B-form DNA is approximately 20 nanometer, 0.34 nanometer, 3.4 nanometer, 2 nanometer. 2 nanometer. Come on, answer this question. Yes. Oops. 1 second. I'll just. Yes. What's the answer for this? Yes. Very good. Very good. 0. One turn of a helix in a B form. Students, helix kelidare. Helix or pitch of the DNA kelidare. They're not asking one turn how many base pairs. They're asking one turn of helix or the pitch of the DNA. Pitch of the DNA. So answer now. Answer now. No. Madesh, think once again. Think once again. This one, what you're saying is the length of the DNA. Okay, length of the DNA. That is different. Length of the DNA approximately 2.2 meter. Yes, very good. It's option number 3. It's option number 3. 3.4 nanometer. Okay, 3.4 nanometer. Just now I taught you know pitch of the helix or helix word bandre automatically 3.4 nanometer ante irbeku. So 20 marks. Okay, 20 marks. So whoever got 20 marks, 16 marks, 12 marks, 4 marks, please drop it down in the comments. Okay, I want to see all of your comments. So next class I'll be doing packaging of DNA. Now students, let me just show you. Okay, this is the channel that you're watching, Vedantu Karnataka channel. This channel will click madre. Okay, just click on this channel. And if you go to videos, let's say you're preparing for next year, okay, for KCET, I have already made a 1-year strategy, okay, for KCET, 1-year strategy. And if you go to live students, we have started taking classes called Mission MBBS. So one class has happened yesterday, Principles of Inheritance, and here one more class has happened today, Molecular Basis of Inheritance. ಇಲ್ಲಿ ನೋಡಿ KCT class, okay, Ugram batch is happening for KCT. So ಇದರದೆಲ್ಲ notes ಬಂದ್ಬಿಟ್ಟು ನಿಮಗೆ ಒಂದು ಎಕ್ಸೆಲ್ ಶೀಟಲ್ಲಿ ಕೊಡ್ತೀವಿ. ಸೊ ಟಿಲ್ ದಟ್ ಯು ಹ್ಯಾವ್ ಟು ವೇಟ್, ಅದೊಂದು ಸ್ವಲ್ಪ ಲಾಂಗ್ ಪ್ರೊಸೆಸ್, ಅದೆಲ್ಲ ಆರ್ಡರಲ್ಲಿ ಮಾಡಬೇಕು. ಸೊ ಕಂಪ್ಲೀಟ್ ಆಗಿ ಒಂದು ನ್ಯೂ ಪ್ಲೇ ಲಿಸ್ಟ್ ಇಲ್ಲಿ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಇವಾಗ FA1 ಪ್ಲೇ ಲಿಸ್ಟ್ ನೋಡ್ತಾ ಇದ್ದೀರಲ್ಲ, ಆ ಥರ ಮಿಷನ್ MBBS ಅಂತ ಒಂದು ಸಪ್ರೇಟ್ ಪ್ಲೇ ಲಿಸ್ಟ್ ಓಪನ್ ಆಗುತ್ತೆ. ಸೊ ಆ ಪ್ಲೇ ಲಿಸ್ಟಲ್ಲಿ ಎಲ್ಲ ಚಾಪ್ಟರ್ಸ್ದು ಪಾರ್ಟ್ 1, ಪಾರ್ಟ್ 2, ಪಾರ್ಟ್ 3 ಕಂಟಿನ್ಯೂ ಆಗುತ್ತೆ. ನಾಳೆ ನಾನು ಪಾರ್ಟ್ 2 ಮಾಡ್ತೀನಿ. ಸೊ ಹೂಎವರ್ ಹ್ಯಾಸ್ ಜಾಯಿನ್ ಟುಡೇ, ಹೂಎವರ್ ಇಸ್ ಪ್ಲಾನಿಂಗ್ ಟು ಜಾಯಿನ್ ಟುಮಾರೋ, ಪ್ಲೀಸ್ ಜಾಯಿನ್ ಮಿ ಅಟ್ 5 o'clock ಟುಮಾರೋ. ಐ ವಿಲ್ ಬಿ ಡೂಯಿಂಗ್ ಪ್ಯಾಕೇಜಿಂಗ್ ಆಫ್ DNA, ಆಮೇಲೆ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಇನ್ DNA. ಓಕೆ, ನನ್ನ ಲೆಕ್ಚರ್ ಸ್ವಲ್ಪ ಲಾಂಗ್ ಆಗಿರುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್, ಬಿಕಾಸ್ ಚಾಪ್ಟರ್ ಸ್ವಲ್ಪ ದೊಡ್ಡ ಚಾಪ್ಟರ್. ನಾನು 1 ಅವರಲ್ಲಿ ಮಾಡೋಕ್ಕೆ ಟ್ರೈ ಮಾಡಿದ್ರೆ ಇಟ್ ವಿಲ್ ಗೋ ಟು 7-8 ಪಾರ್ಟ್ಸ್. ಸೊ ಯು ವಿಲ್ ಆಲ್ಸೋ ಬಿ ಬೋರ್ಡ್, ಐ ವಿಲ್ ಆಲ್ಸೋ ಬಿ ಬೋರ್ಡ್. ಸೊ ಅದಕ್ಕೆ ಒಂದು ಸ್ವಲ್ಪ ಲಾಂಗ್ ಲೆಕ್ಚರ್ ಮಾಡಿದ್ರೆ it will be more easy to complete the chapter faster. Okay, because adamele ನಾನು 11th simultaneously start ಮಾಡ್ತೀನಿ. 11th allo high weighted chapters ಇಂದ start ಮಾಡ್ತೀನಿ. Okay na? So yes, let's crack NEET this year. Okay, and let's support each other. I will support you in learning the chapters. You support me by sharing the video with all your friends. So students, see you all in next class. Till then, take care. Bye bye. So I hope all of you enjoyed the class. Enjoy ಮಾಡಿದ್ರೆ please let me know in the comments how the class was, and also let me know your marks. Okay, bye bye.